Good morning, Kunal. Can you hear me?
Yes, ma'am.
Kunal, can you hear me?
Yes, ma'am.
Yes, madam. Your voice is audible. Kunal sir, yes, we can start today's session. Madam has already joined.
Okay, sir.
The times are changing, and so is the world around you. At CVM University, we help you prepare for the future, like we have with more than 10 lakh students. Come, join the educational legacy of 75 years. CVM University, enlightening the future generations.
Good morning to all of you present over here. I, Krunal Solanki, Assistant Professor, Department of Pharmaceutical Chemistry and Analysis, IICP would like to welcome all of you on the second day of online 2 days refresher course. Start this second day with the quote of Terry Gulmetz that I like my coffee black and my morning bright. For making a bright morning, you must have knowledge about your profession. And for that, we have gathered for gaining some knowledge regarding our pharmacy professions from different experts. Moving towards our first plenary session, we have with us Dr. Nazneen Surti ma'am. I request Ms. Jinal Tandel ma'am, Assistant Professor, Department of Pharmaceutical Chemistry and Analysis, IICP, to introduce Dr. Nazneen Surti ma'am.
Very good morning to everyone. Dr. Nazneen Madam has about total 23 years of experience, 18 years in academia and 5 years in pharmaceutical industry. Main area of specialization are pharmaceutics and novel drug delivery system. 3 PhD students and 54 PG students completed their research work under her guidance. She has published 41 research papers in national and international journal of repute. Total 6 patents are credited under her name. Among them, 2 patents are granted. She has published total 6 book chapters. Madam has organized 20 different seminar, workshop and conference and total 2 government granted projects handled by her. Different 6 awards were credited to her and so many times invited to deliver the lecture in various events. She is life member of IPA, MPA, APTI, APP and also editorial board member of World Journal of Pharmacy and Pharmaceutical Science. Today, Madam is going to deliver a talk on medication adherence. On behalf of IICP College of Pharmacy, I, Jinal Tandel, welcome you, ma'am.
Very good morning to everybody. And thank you, Jinal Madam, for such a nice introduction in the morning. So I am Dr. Nazneen Surti. I work as a professor with IICP College of Pharmacy. And today I am going to deliver a talk at the refresher course for registered pharmacists organized by IICP College of Pharmacy. Is my presentation visible?
Yes, ma'am, it's visible.
Okay. So before starting my talk, I would like to thank the course director and the principal of IICP College of Pharmacy, Dr. Harsha Patel, the coordinators of this refresher course, Dr. Dimal Shah and Dr. Usman Ghani Chalotia, for inviting me to deliver a talk at this refresher course. Today I'll talk something about medication adherence. We know what all it is, and by adhering to a medication, how we can enhance the patient's health and outcome of a therapy. I'd like to brief upon that. So this is what I'm going to talk about: adherence concepts and terminology, some statistics related to medication adherence, consequences of non-adherence, factors affecting medication adherence, prerequisites for adherence, strategies how we can improve medication adherence, and what are the tools which help patients for adhering to their medication regimen. So we all know what medical adherence is, right? The patient, if the patient takes the right dose of medicine at the right time, in the right way, and for the correct period of time, then it is called as he is adhering to his medication regimen. World Health Organization defines medical adherence as the degree to which the person's behavior corresponds with the agreed recommendations from a healthcare provider. If a healthcare provider has told you to take a medication in a certain way for a certain period of time, then you have to adhere to it. Then only there will be health outcomes. Otherwise, they may lead to worsen your condition. So we all rely upon medicines, right? Either to cure or prevent diseases. We all know that it is very important. So medication adherence is actually the most important point in a medication regimen or therapeutic regimen. Still, this is the only point where we are weak. This is the weakest link in our healthcare system. And we as pharmacists have to strengthen this link so that it improves the overall health of the nation. So medication adherence becomes very important. It is important for everyone, right? But it is very important when we are dealing with chronic diseases where the drugs have to be taken for a longer period of time, and diseases where serum drug concentration should be maintained. There are some potent drugs which require, and some essential drugs like anti-epileptics, where the drug concentration needs to be maintained or a narrow range, right? You cannot miss a dose. The drug concentration then will go down and the effect of the drug will not be seen. Or medication adherence is also very important where diseases affect the general health of the public, like when there are some contagious diseases going on, you have to adhere to the medication so that you don't pass on the disease to other people. Then medical adherence is also important for replacement therapy, like hormones therapy, especially insulin plus other hormones, which our health is very dependent upon. Okay, so medication adherence is most important on all the ages. I mean, like even patients, all the ages, for everybody, medication adherence is important. But it is more important for pediatric as well as geriatric patients where they need help to adhere to their medication schedules. So these are certain terminologies which tell you the behavior of a patient towards his medication. So the three terms are used: compliance, adherence, and persistence. Compliance is now disfavored, and compliance is not used much because it suggests that a patient passively follows the doctor's order. He is not really interested, but he is complying. He is taking the medication just because his doctor has told him to. Adherence shows that the patient is willing. He understands his diagnosis and he is willing to take the medication, and he believes in the therapy. Persistence is the ability of a person to continue taking medication, and this persistence means being there for a long period of time. Sometimes the therapy may last for months or years or even for a whole lifetime that a patient lives. So these are the three terminologies which are commonly used to describe the behavior of patients during their therapy. These are when do you call it that it is medication non-adherence? What all falls into medication non-adherence behavior? First is to fail to fill the prescription. You go to a doctor when a patient goes to a doctor when he feels something is wrong with him, right? The doctor writes a prescription, but you don't purchase those medicines. You don't go to the pharmacy and purchase those medications. That means you failed to fill the prescription for the first time. Even after you buy the prescription, you skip doses. You don't take all the doses that you've been told to take. Then if you're taken all the doses, if the medicine has to be repeated every 15 months, you don't go and buy those medicines again. You don't refill your prescription as you had been directed by the doctor. Then either you take more medications or less medication than prescribed. If the symptoms are less, you tend to take less medicine. If you've been told to take 3 times in a day, if the symptoms are less, you will take only 1 time or 2 times a day. Or if there are more symptoms, you tend to take more, but that should not be done because what is prescribed has to be followed because there is a reason that it is being prescribed in that manner. Then taking someone else's medication also falls under medication non-adherence. If you have symptoms similar to someone else, you can't take those medications because people are different in how they behave towards different medications, right? We all know that our metabolism varies from person to person. All the ADME factors vary from person to person. So if one medicine is working with one patient, you cannot be sure that it is going to work for you, even if you have the same symptoms. So taking someone else's medication also falls under medication non-adherence. Then taking expired medications, there is an expiration date on your medication beyond which you're not supposed to take the medication, right? Because the medication might have degraded. Then sometimes you're told by the doctors to take certain medicines with food, without food, with some drink or not with some other drink, right? So when you're told to do such a thing, then you have to follow it because there's a reason behind telling you that. Some medicines, the absorption of some medicines it goes down in the presence of certain food. Certain foods interfere with the absorption of some medications. They interact or degrade a particular medication. So those foods have to be avoided. Or you have to take food in case where you're taking a gastroretentive drug delivery system, because those systems require food to be present in your stomach. Then if you store medications in the wrong place, again, they are going to get degraded. So that also falls under non-adherence. So once we know what is non-adherence, then we can find out what are the factors affecting this non-adherent behavior, right? Before that, I'll tell you some statistics related to medication adherence, which has been surveyed over a period of time at different places around the world. So almost 20% of the patient, this in some studies, this 20% is almost 50%, right? Some countries, this goes up to 50%, but this is a general thing which has been put here. So 20 patients don't fill that prescription at all. They don't, after the doctor has written the prescription, they don't go to the pharmacy and buy the medicines at all. Out of the people who buy medicines, 12% of those people, they don't take the medication. They buy it and keep it at home. They don't start taking that medication. Then 29% of the people stop taking the medication before it is finished. You buy 20 tablets, you have been told to take 30 tablets in 10 days, but you stop taking medicines after 20 tablets. So even before the medicines are over, you stop taking that medication. 29% of the people stop taking the medication. And 22% of the patients take less of the medication than is prescribed on the label. So, and in persons with newly diagnosed high blood pressure, if the person is diagnosed with high blood pressure and it is a new diagnosis, then after 12 months, the adherence goes as low as 78%. And after 54 months, the adherence is found to be only 46%. After 54 months, the adherence goes down to 46%. So these are the statistics, overall statistics, which are related to adherence. Now, examples of classes of drugs for which most non-adherence is reported. You can see that all these drugs, antidiabetics, CVS, HIV, and antibiotics, they are all taken for chronic conditions, right? We all know that for antibiotics, we have to take the full course of medicine. If you have been told to take tablets twice in a day for 5 days, then we have to take it for 5 days. If we stop taking after 2 days or 3 days, if our symptoms go away, we stop taking it after 2 days, but it cannot be done, because if you stop taking it before the course is over, then some bacteria may still live in your system and they might again grow. You might get the infection again. And over a period of time, these bacteria might become resistant. Then with HIV medications, the problem is the people who are suffering from HIV, they have to take multiple drugs. The side effects of those drugs are also high. So we can see that adherence is also less with HIV drugs. Then drugs like antihypertensive, platelet agents, and statins. Sometimes a person who has hypertension will not feel the symptoms, or antiplatelet agent and statins are taken for symptoms which may develop after a long period of time. So even that people know that if they don't take these medicines, it is ultimately going to increase the risk of myocardial infarction or stroke. Nothing is happening immediately. So these drugs fall into non-adherence. Similarly with anti-diabetics, the diabetic patients don't get symptoms unless the sugar level becomes very high. So they are reluctant of taking medicines because they don't have symptoms, but they even if they know very well that over a period of time, if they don't take anti-diabetics, then they might get diabetic retinopathy, neuropathy, etc., etc. But some people don't know, and we have to make them aware. We have to educate the patients that not taking these medications is ultimately going to harm them. Then similarly, antiplatelet things and clopidogrel and aspirin, they doesn't show any effect as such, right? But they do help with your non-deposition of plaque in your arteries. This is medication adherence statistics reported by scientific reports in 2020. Here you can see the level of adherence, percentage adherence, and here you can see the drugs. So AC inhibitors like captopril, then P2Y12 receptor inhibitors like aspirin and statins. So this is the result for the first quarter, adherence for the first quarter. As the time progresses, the adherence goes down, right? Up to the fourth quarter, it reduces in all the classes of medicines. These are the different classes of drugs, and these are the adherence, percentage adherence of patients after first quarter, second quarter, third quarter, and fourth quarter. If you consider this as 3 months or 4 months, then it goes down after 12 months. This is some of the statistics of antihypertensive medications taken by people. So high adherence, people adhere to antihypertensive medicines, percentage is only 16.2, whereas medium adherence is 47 and low adherence is 36.5. The other conditions where we can see non-adherence rates, high non-adherence rates are asthma. 25% non-adherence of up to 25 to 75% can be seen as asthmatic patients. Asthmatic patients, they take medications only when they feel the symptoms, when they are feeling breathless and all. They don't take preventive medications. They don't take the medications that's prescribed. They don't take medication when they don't have any symptoms. Similarly, with major depression, schizophrenia, and anxiety disorder, these are all mental disorders where the patients have to be helped. They need help for adhering to their medication schedules. Again, there's a difference between adherence when we talk about males and females. In a study, they divided people into high and moderate adherence people and with low adherence people. In both the groups with high adherence and low adherence, percentage of patients adhering was high, and with males, number of males adhering was also high, and number of males non-adhering was also high in low medical adherence. But generally it is seen or it is observed in other studies that females are more adherent than males to their medications. Then what happens if you do not adhere to your medication schedule, right? If you do not take if people do not take the medications properly in the manner that they have been told, then the disease might progress. Instead of the disease getting cured, disease will get cured only if you take medicines properly. So if you are not taking medicines properly, then the disease will progress. Your disease is going to worsen, which will ultimately lead to increased hospital admissions. And hospital admissions will again increase the medical cost, also non-medical cost. It will increase mortality. The death rate is going to get increased because sometimes the disease progresses so much that then it is not controlled, that it doesn't come under control. So people die. If they don't die, at least there's a low quality of life they have to live with. If the disease progresses, then the patient is going to lose some of the functions, vital functions of life, and ultimately lead to low quality of life. And if you don't take medication, ultimately the medication which you have purchased is going to go waste, right? And ultimately it is going to affect the cost. These are again some statistics. How much hospitalization has occurred with diseases like CHF, diabetes, hypertension, and hypercholesterolemia. So you can see that if adherence increases, the percentage of adherence increases, the hospitalization goes down. If you compare 1 to 19% adherence with 80 to 100% adherence, you can see that the number of hospitalization have gone down considerably with diabetes, hypertension, and hypercholesterolemia. With CHF, it might not have been that different, but with diabetes, hypertension, and hypercholesterolemia, the the incidence of hospitalization has gone down considerably when the adherence increased to 100%. So talking about the factors affecting medication adherence, once we know these are the factors these are the 5 categories which are given by WHO. WHO has divided the factors affecting medication adherence into these 5 broad things: socio-demographic factors, therapy-related factors, health system-related factors, condition-related factors, and patient-related factors. Okay, socio-demographic means something related to the age of person, the age, the race, the ethnic group, the culture that he belongs to, the gender, the marital status, the income, the education, the occupation. All these comes under the socio-demographic factors. So under socio-demographic factors, as I say, the age becomes a factor. So whether a person is very young, whether pediatric and geriatric patients are there, that will affect their medication adherence. Then if person is literate or not, whether he is educated, whether he can understand his diagnosis and whether he can understand the therapy-related outcomes. So, and whether he belongs to a stable home, whether he is a survivor of war or disaster, whether he belongs to a certain social group or not. Then whether the patient belongs to a minority. The cultural differences also matter very much, right? If some cultural or if some religion believes that certain things cannot be eaten, for example, gelatin comes from gelatin is obtained from animal tissues, right? So some people who are vegetarians, they are reluctant to take hard gelatin capsules. So for such beliefs, what can be done is changing the empty gelatin capsules to capsules which have been made for HPMs. Another factor that affects the adherence is if low income, like if the person cannot afford the therapy, how is going to comply or how is going to adhere, and especially for therapies which take a long time. Another things are patient-related factors, right? So again, low literacy here is a main thing. Then patients are not adherent simply because they forget to take, right? Forgetfulness. Suppose somebody is going to office, he has to take medication in the morning and he forgets because he's running around. So simply he forgets. So medication adherence is not always intentional. There are 2 types of medication adherence, intentional and unintentional. So half of these things fall into unintentional and half fall into intentional. So a patient just forgets to take the medicine because he's rushing to the office or so many other reasons, or there's a lack of knowledge. There's a lack of knowledge. He is not being made aware of non-adherence. Then people believe in alternative therapies. Some people, if they are prescribed, like they'll go for alternative therapies, or homeopathy, Ayurvedic, instead of taking the mainstream medicines. Then they might not go to the doctor for follow-up. Certain diseases require constant monitoring and constant visits to the doctors, but certain people, certain patients, they just do not go for follow-ups. Patients then do not take medicine because of adverse effects, right? Sometimes they get adverse effects, and because of that fear, they will not take the medicines. Then some people are just reluctant. They go on a denial mode, right? They deny that I have a particular type of disease and I don't need this medicine. So it is belief related. Other factors are therapy related. Okay, how complex the regimens are, right? Sometimes the patient has to take multiple drugs for 3 times a day, right? If you can make that multiple regimens into simple regimens, like if a person has to take a medicine 3 times a day, so instead of giving him 3 times a day, if you can give us a stain release medication or an implant or a depot type of preparation which releases drug over maybe 8 hours, 12 hours, or even months. In hormonal therapy, you can give an implant to the patient where They release the drug over months, right? So instead of taking a pill every day, if there's an implant or if there's a transdermal system which releases the drug for a long period, that will simplify the regimen, right? So another factor is time to benefit. If the patients generally want to see the effect immediately, so if the time taken for the medicine to show the benefit is long, then also they will get impatient. They'll stop the therapy. Right? And the duration of treatment, same thing, time to benefit and duration of treatment. If the duration of treatment is less, then they are more adherent than. So in generally acute conditions, there is more adherence than chronic conditions. Then other factors are condition related, right? If a person has multiple chronic conditions, for example, if he has multiple diseases, he's taking multiple medications. So that affects adherence. Symptom severity. Okay, if there are no symptoms, then the person will not remember to take the medicine. Right? If so, adherence is more if the symptoms are persistent, or you have symptoms, then you will remember to take the medicines. If there are no symptoms, you will not remember. So, and if the person has depression, psychosis, and dementia, he is especially prone to non-adherence, and that person has to be reminded by caregivers or family to take the medication. Other major factor affecting non-adherence is the healthcare system. How is your relation with your doctor? Whether you are made aware of your therapy or diagnosis, whether you trust your doctor or not. Whether it has hospitals that you go to, whether it has enough, whether it is patient-centered, it has enough physicians to take care of you, whether they are providing you with the proper knowledge, and whether you access. I mean, like sometimes waiting periods, like simple as that. If you have to wait for a doctor for a long period of time, for long hours, then also that leads to non-adherence sometimes. So once you know what are the factors which affect medication adherence, then you can design the strategies to improve this. We know these factors, right? Now we can overcome these hurdles and do things so that these things get nullified and adherence increases. So adherence requires that a person shows interest in her health. Like compliance is only when a person passively follows the doctor, but adherence requires that the person who is taking medications shows interest in her health, in his or her health, and he understands the diagnosis, believes that the prescribed treatment will help, know exactly how to take the medication and how long this therapy is going to last. Then he finds ways to fit the medication regimen into his or her daily routine. I mean, like he ties his time of taking medication to his daily routine, taking this before tea, or taking this after food, or taking this after a walk, etc., etc. And the person should believe that he can carry out the treatment plan. Right? You have to make him believe that whatever you are doing is going to help you, make you treat your disease. And the person should trust the healthcare practitioners. He should believe that the healthcare practitioners, whatever they are doing for his health, and they are going to treat his disease. So based on the factors, again, these are some of the strategies by which we can improve medication adherence. The first is you can simplify the treatment regimen. As I told you, you can reduce the number of daily doses and pills wherever possible by using combination medications. If he has to take 2 antihypertensive drugs, then there are combinations available where they can combine these 2 drugs, or you can give a sustained release medication, or you can give a transdermal patch instead of oral medications, or a depot, so that he doesn't have to take more number of doses and that frequently. Then patient education can be done. Patient counseling is very important. Patient counseling is very important. You counsel the patient. You provide knowledge to the patient. To improve his health literacy, right? You ask the patient what is going to be convenient to him. You engage the patient in decision making. tell the patient, see that this is your condition, and let us decide how we can design the regimen so it is most comfortable to you. So you can engage in shared decision making with the patients. Then you can use of technology and reminder. There are so many technologies and reminders available, which I'll be discussing in the coming slides, which help remind the patient to take their medications. Then there should be regular follow-up and support. You can schedule frequent appointments to monitor progress and address the issues, and establish a patient-centered relationship to build trust and accountability. Then sometimes the pharmacist gives incentives and motivate the patients, like if they come for regular refilling of the prescription, they give discounts for maintaining adherence. And they also motivate the patients by setting personal goals, like you walk so many steps in a day, or if you do this thing, then this will happen. You motivate them. Or you address the healthcare system barriers. If there are some healthcare system barriers, one of the factors which affects, so you can address those barriers by collaborating with healthcare providers to improve communication and coordination, or you can develop protocols for medication, better protocols for medication management. Then if there is cultural sensitivity or diversity, some people due to religious beliefs, they don't take some medicines, right? Because they are made up of animal substances and all. So there was a concern about, this similar concern when vaccines were prescribed for corona, right? Some people believe they use substances which are not allowed in their religion to take. So they were not taking those vaccines. So in that, you have to involve the community leaders, or whoever community leaders are there. For such people, you have to involve them. You have to convince them so that they can convince their people and bridge the communication gaps. Plus, you have to tailor interventions to suit. You have to do something which suits their cultural beliefs and practices. And you have to engage the family and caregivers for geriatric patients and pediatric patients and mental health patients, with the family members and caregivers that play a very important role in medication, in adhering to their medication, right? So you have to educate them about the treatment plan and involve them in the process of setting the goals. So what the pharmacist can do for increasing adherence is very simple, right? So simplify the regimen, impart knowledge, simple it is, okay? Simplify the regimen, impart knowledge, modify patients' beliefs and human behavior, provide communication and trust, leave the bias, and evaluate adherence. Now, how do you evaluate adherence? You have done everything. You educated the patients, you improved the healthcare system, everything. But then how you have to evaluate whether what you have done is increasing the medication adherence or not. So there are different measures there are different tools to measure medication adherence. Some direct methods are there, some indirect methods are there. And there is a Morrisky medication adherence scale from which you can determine whether a patient is adhering or not. Indirect methods, we have like checking the blood of the patients for the presence of medication or some metabolite or some biological marker which is present in the blood. So this is a direct method, which is a kind of invasive because you have to take the patient's blood. So this is not recommended, or maybe this is recommended in some diseases. Indirect methods are there where there are patient questionnaires. You give questionnaires to the patients which they fill out. Then patient self-reports are there. The patient writes the reports himself. Then there are pill counters, how many times the prescription is refilled. Then you can assess the patient's clinical response. The clinical response, or for example, you measure the blood pressure, etc., etc. And then you find out whether he's taking his BP medications or not. And there are some electronic medication monitors, which I'll discuss later. And there is Morrisky medication scale, which tells you whether the behavior is intentional or unintentional. So what is this Morrisky medication scale? This is the medication scale where the patient is asked 8 questions. Questions 1 to 7, they have to answer whether it is yes or no. Right? 1 to 7, they'll ask yes or no. When he answers no, he gets 1 point. For all the questions except question 5, a no will get 1 mark. In question 5, a yes will get 1 mark, right? So question 1 to 7, you have to answer yes or no, where no gets 1 mark. For all the questions except question 5, where question 5 gets 1 mark for yes. For the 8th question, you have, this is a Likert scale, okay? This is the Likert scale where you have to rank this. If you take this, you may get a score of 4, 4, 3, 2, 0, and 1. So whatever the patient takes, based on that, he will get either 4, 3, 2, 0, or 1 mark. So whatever marks he gets here is then divided by 4. Then addition of all these marks gives you a number. The highest score in this scale, what you get is 8. So if you get the score of around 7 or 8 or 6 to 8, then you can say that the patient is adhering to the medication. Less than 6 score is non-adherent to the medication. Then there are medication adherence tools, right? There are so many medication adherence tools available in the market now, which help the patient remember to take the medications, right? So some of these are listed here. We have pill organizer. This is a kind of a pill organizer, picture of a pill organizer, where the pills are put in the boxes named by the days of the week, Sunday to Saturday. Similarly, on strips, we also have, if the person has to take medication for 21 days or for the whole month, then the dates are written on the strips. So the patient does not forget, like if he sees these things or the strips on which the number of days are written, then he can immediately come to know whether he has taken or he has missed the dose of that particular day or not. Then there are various reminders that are available in the market now. Example, these are some of the medication reminders which are available in the market now, where we have a tab timer. This is like a tablet, okay, where you can put, this is like an alarm system where you can put alarms which go off or go on every 8 hours or every 6 hours whenever you have to take the medicine. So it will send an alarm and remember that you have to take the medication. This is the e-pill time cap and bottle, where the cap of the bottle has the time. It tells you the time when the bottle was last opened. So by if 12 hours has passed when you last opened the bottle, that means you can find out, you can come to know how many doses you have missed or whether you have missed any dose or not. Here the cap has the timer. Here it is a kind of alarm. Then there's a pill pack by online pharmacies. This is a you know what is Amazon, right? So if you subscribe to this pill pack, they will deliver the pre-ordered or pre-dose medicines to you. So this does not give you a daily reminder like these do. These two things give you daily reminders, but this does not give you a daily reminder. They will automatically fill your prescription and deliver it to you free if you subscribe to this pill pack. Then MediSafe is a mobile app. It's an app which you can download on your mobile. You can put down all the information about you, how many medicines you take at what time and how long you're supposed to take. So they will send you alerts. They will send you either SMS messages or whatever alerts they have to, so that you can get up and take your medicines at that particular time. Sometimes these are free, and sometimes you have to take the subscription so that they can offer more services. And these things are not linked to any pharmacy, but this mobile app is also linked to a pharmacy. So you get advice from the pharmacist regarding some of the treatments. And this is a kind of a pill dispenser, right? This is connected to the Wi-Fi of your house, and there is a sensor which sends signals, right? So whenever it is time for you to take the medicine, this dispenser is going to dispense the medicine in this cup. After dispensing the medicine in this cup, it will send a signal to you through the Wi-Fi of your house. So you just have to come to the dispenser, take whatever is dispensed, and then take the medicine. This is kind of a costly thing. Other things are not so costly and can be used by patients. Some more medication adherence tools are mHealth, eHealth, and eMMDS. So mHealth is an abbreviation for mobile health. Again, it is an app So patients are able to record their treatment schedules. They can store and monitor their health records on their personal mobile devices. And it was surprising for me to know that there are over 318,000 health apps available in the market from which you can choose. And popular examples of these mHealth include Fitbit, which many of us now use, Google Fit, and Apple Heart Study. E-health, it's a much broader understanding than mHealth, and then this technology used the technology used here. So technology used to improve healthcare practices with e-health, they include electronic health records, the health record of patients, then patient administration systems, lab systems, and other records. So all your lab records are also stored in this e-health system, which is much broader than the mobile health system. So things which cannot be stored in your mobile health application can be stored in e-health, right? And then last we have eMMDs. These eMMDs use audio and visual signals to prompt the patient when they have to take medication, dispense medication, and gives instruction to the patient, or they can contact the caregiver or your family member in case whenever it is needed. So these are the apps and some tools which are in the market for people to use who cannot adhere to the medication because just because they don't remember, right? It is unintentional. When it is intentional, it cannot be cured by these. But when it is unintentional, we just forget, then you can use these things. These are some of the companies which are there in the market today who have come up with all these medication adherence tools, and they are doing very well. How well they are doing and how well they will do can be seen from this global medication adherence market by value. The value is the global medication market is growing very much. Like this is the data for period from 2018 to 2028, US $1 million. So in 2021, the global market for medication adherence tool was $2,668.1 million US, which is predicted to increase so much. I cannot see this. Which is predicted to increase up to 5,562.3 in about 2028. So the market for this adherence tools is predicted to increase so much around 2028, up to 2028. This also indirectly tells you that how the problem of medication adherence is increasing day by day in the world, right? And how we really need the tools so that people can adhere to their medication schedules and improve the quality of their life or improve the diseases and contribute to the overall health of the nation. So here I conclude. So medications generally do not work in patients who do not take them. We have to take the medications properly so that they help treat us. So medication adherence is crucial in achieving positive health outcomes. And by addressing barriers and implementing effective strategies, we know the barriers, so hence we can implement effective strategies. We can enhance patient adherence and the overall well-being of human beings. Thank you. I'd like to take any questions if there are.
Thank you, ma'am. But there is no questions for. Thank you, ma'am, for sharing this wonderful knowledge with us. And there is no questions from the participants.
Okay.
Okay, thank you, ma'am. An investment in the knowledge pays the best interest. After completion of the first plenary session, I would like to welcome Dr. Ankita Bhavsar, ma'am, Assistant Professor, Department of Pharmaceutics, IICP, to introduce our next expert, Dr. Nikunj Parekh sir for delivering his knowledge on drug discovery and development. Over to you, ma'am.
Thank you so much, Kunal sir. Am I audible, sir?
Yes, madam. Yes, ma'am.
Okay, thank you, sir. So as further proceeding towards our refresher course, I would like to introduce our most eminent speaker of the next session, and he is Mr. Nikunj Parekh. He is an assistant professor as pharmaceutical chemistry and analysis department of Ramanbhai Patel College of Pharmacy, Charuset, Changa. Moreover, Nikunj sir is a lab manager of the Sophisticated Instrument Laboratory, RPCP. He completed his B.Pharm from Indukaka Ipkowala College of Pharmacy, New Vidyanagar in the year of 2014, and he is M.Pharm from the Faculty of Pharmacy, the MS University, Baroda in the year 2016. He initiated his career as R&D scientist, Medicinal Chemistry Department of Torrent Research Center as a R&D scientist. Medicinal Chemistry Department of Torrent Research from Torrent Research Center, Gandhinagar. He has expertise in molecular designing synthesis route, chemical synthesis, chromatographic separation, API process development and optimization, as well as impurity profiling and toxicity studies. He has successfully guided 2 M.Pharm QA students and 8 B.Pharm students for their research projects. Currently, he is pursuing his PhD from Charuset University, and his area of research is design, synthesis, and biological evaluations of chemical moieties for prevention of cancer metastasis. I would like to Nikunj sir to illustrate some of his knowledge to all the attendees of the refresher course. Thank you very much, sir. Over to you, Nikunj sir.
Thank you, ma'am. Hope my presentation is visible.
Yes, yes, it is visible.
Yes, okay. So, very good morning, all of you, dear participants, and pranam to all my seniors present over here. And thank you very much, ma'am, for a nice introduction. It's a beautiful Sunday morning to discuss about such a wonderful topic, that is the drug discovery and the development. We are 1 hour together to discuss upon the journey of any particular chemical. Okay, so to become a drug. So let's start our journey. So today we discuss about how basically the drug discovery starts, what is the molecular modeling, which are the in silico tools which is used to design any particular drug, then how to synthesize that particular drug, what are the consequences related to that drug discovery, preclinical studies, it means how that drug that affects the biological system, then the cellular system, how that drug binds with the receptor, everything in the animal models, that is the preclinical study. Then we discuss about the human volunteers, the drug trials on the human volunteers, that is the clinical trials. Then the reviewing whatever the data that is collected in the step 1, step 2, and step 3, that is all we need to submit to the regulatory agencies, and then they give us a marketing rights, marketing of that particular formulation. And then the 5th stage of the drug discovery and development is the continuous monitoring and taking the feedback from the patients and doctors. So this is whole drug discovery and development steps. In this particular talk, whenever you see such, just a minute, whenever you see such, we can say the icon, it means I want to tell you something. I have some experiences in the drug discovery in the Torrent Research Center. So I have something to tell you. So whenever you see this particular icon, so that is something. Then if you find something interesting, something new in this particular presentation, you just need to take a screenshot for that. Okay, so that may help you. And whenever you like something, so give in a chat box yes. Okay, so we proceed further. So very first thing, why we need drug discovery? What is the importance of drug discovery? Okay, so very first thing of the drug discovery is we need to reduce the cost of drug. Okay, drugs should be affordable to all, each and everyone. Okay, it should be easily available to each and everyone. There is a strong and we can say the effective supply chain with the minimum cost of any particular drug. That's why we need a drug discovery. Another thing, another objective for the drug discovery is the pandemic and epidemic, what we face. We know every 100 years there was one pandemic, and every 10 years there is an epidemic. We know, we heard about the Spanish flu that occurred in the 1920. We know about the plague and influenza, that is the epidemics in 1960. We know about the Hong Kong flu, it is in 1970. 1970s, HIV AIDS in 1980, and we all faced the pandemic of COVID-19 in 2020. So pandemic, epidemics, રોગચાળો કહીએ, pandemics, જેને આપણે મહામારી કહીએ, each and every in this particular pandemic we need drugs, okay, to save our lives, to save the life of our relatives, to save the life of society. We know the importance of remdesivir and amphotericin B during the COVID-19. If it is not there, what happened? Just think about that. So this is the importance of the drug discovery. Another thing is the drug resistance. I need something to tell you. Drug resistance is the biggest problem nowadays. Biggest problem nowadays. There is a one news article in which they just survey that there is a 7 lakhs death per year because of the drug resistance. 7 lakhs death per year, and that may be increased up to 10 million death per year in 2050. 2050 સુધીમાં આ આંકડો 10 million સુધી પહોંચી શકે because of the drug resistance. Okay, so how severe this drug resistance is? I have something to share with you. This is one report. This is one report, that is the culture report. One patient is suffering from the urinary tract infection, UTI. એનાથી સફર થતા હતા patient, and this is recent report just in December 2022. Okay, so you can see this is the list of drugs. You can zoom also in your screen. This is a list of drugs, and see the sensitivity of antibiotics. That the sensitivity of antibiotics towards that particular bacterium. 40% of antibiotics are, we can say, the resist to that particular, we can say the bacteria that cause the UTI. Only 60 to 65% antibiotics are effective. Whenever you see this S, it means sensitive, and whenever you see R, it means resist. It means that patient is resist from that particular antibiotics, and antibiotics are the lifeline. If there is no antibiotic, if antibiotics are not causing any effect, there is an end of life. Okay, so this is why we need drug discovery. There is a one news article in which they declared that after 1990, 1990 પછી only there is only three class of antibiotics are invented. So we are just lacking in the discovery. So this is the importance. Okay, another thing is we need to treat the previous untreated conditions like cancer. There are some rare diseases. We need drug for that. We don't know the exact cause of cancer. There are so many factors that cause the cancer. ઘણા બધા ફેક્ટર્સ છે જે કેન્સરને માટે જવાબદાર છે. કોઈ એક પર્ટિક્યુલર વે કાં તો કોઈ એક પર્ટિક્યુલર ઇટિયોલોજી આપણને હજુ પણ ખબર નથી. એટલું વિગ્રસલી કેન્સર સ્પ્રેડ થાય છે. So we need each and every time we need some new drugs to treat the cancer. We also need the drug discovery because of there is a different metabolism in the human bodies. Male to female, there is a different metabolism. Child to adult, there is a different types of metabolism. So not a single drug is effective in each and every one. We need we can say the series of drugs to treat that particular patient. And last thing is the quality of life. Okay, we all live long. We all need our family members. We all need healthy life. That's why we need drugs for that. So that's why the drug discovery is important. You can see here is the drug discovery market worldwide by the segments in the year 2016, and there is a predicted segment that is 2025. So you can see there is 27.1 billion US dollars is invested in the small molecule drug discovery in 2060, near past. But that particular amount will increase double in 2025. So this is some statistics which indicate how much the mankind is investing in the drug discovery to save their lives. And look at the, we can say, the predicted values, all these predicted values, okay, like the investment in the biologicals and drug discovery. This is overall. So in 2025, it will increase up to $71 billion of investment in the drug discovery. Now there is another question: how basically the companies spend for the drug discovery? How they predict કે આટલું આની અંદર આપણે ઇન્વેસ્ટ કરવું પડશે. So very first thing, spending in the R&Ds and the introduction of the new drug have both increased in the past 2 decades. In 2090, the pharmaceutical industry spends about $83 billion in R&D. And to adjust for the inflation, that amount is 10 times more what the industry spent in 1980s. 1980 માં જે ખર્ચ કરતા હતા એના કરતા 2019 ની અંદર એ ખર્ચ R&D પાછળ એ લોકો 10 ગણો વધાર્યો છે. Between 2010 and 2019, the number of new drugs approved for scale by 60% increase compared with the previous decade, with the peak of 59 new drugs approved in recently in 2018. 2018 માં recently 60 જેટલી નવી drugs approved થઈ છે. This is the figures by which you can predict how the R&Ds are, we can see what is the outcome of R&Ds in last 2 decades. How basically the companies invest into the R&Ds in the drug discovery. So the amount of money that drug companies devote to the R&D, that is depend upon the 3 factors. The government policies, how liberal the government or federal policies is, very first thing. Another thing is the expected cost of developing that particular drug. And the third thing is the amount of revenue that what they expect from that to earn. કેટલો નફો એમાંથી મળશે એ પણ ધ્યાનમાં લેવાય છે. So you can see nearly 360 to 574 crore Indian national rupees that is required for just we can say the complete overall drug discovery. That is same as the GDP of African countries. That is same as the GDP of African countries, and more than 10,000 staff that is involved in the drug discovery. Not only that, 12 to 15, we can say the years that is required to go for a drug discovery. Regarding the research, total 15 to 20 departments are involved to facilitate the drug discovery. You can see how complex the drug discovery is. Here is some statistics, some general idea about the cost, duration, and the probability of attrition of the drug discovery. Everything is depend upon the results and the trials and the probability statistics based upon the results and the whatever they gain after the research. You can see the most dangerous thing over here is this: probability of attrition, drop out of project anytime. You start drug discovery, there is a danger that comes with you. You can see there is a 49% chances of project drop. You may fail in the, we can say in the drug synthesis part. In preclinical part, there is a chances of 31% you fail. You can also see 56% chance. There is a half chance to fail in phase 2 clinical trials. So this is what we can say called as a death valley. You can fail anytime. You should be prepared for that in the drug discovery. So if the data which is transferred from the one state to another state, you should have some belief on that particular data. You just transfer the data to your another partners and then they start their journey. This is the relay race. Basically, this is the relay race, but there is a more probability of the, we can say, the project failure. You can see over here, this graph basically indicate that the patent period, when the patent is granted for 20 years, The graph shows the actual and predicted probability of failure of drug discovery. You can see that after 8 to 10 years of initiation of your research, after granting of your patent, you have 50% chance to fail, 50% chance to fail. And you can predict exactly after 18 years, exactly after 18 years, look the figures, predicted and actual both are the same. There is a, we can say the more than 50% chance to fail in the drug discovery. So overall, the drug discovery is very complex project, same as the search for a life in a universe other than the Earth. It is same like that. Okay. So it requires so much of patience, ઘણી બધી ધીરજની જરૂર પડે, working force, manpowerની જરૂર પડે, intelligence, smart work, and resources and lots of money to invest in the failures. Lots of money you require for a drug discovery. So do you know how many molecules are gifted by the India? How many molecules? કયા નવા નવા કોઈ એકાદી એવી drug કે જે ઇન્ડિયાએ વર્લ્ડને gift kari hoy? Anyone that knows, just write in your chat box so the panelist and the host can see. I will give you 5 to 10 seconds. Just think about that કે કોઈ તમને એવું કોઈ drug ખબર હોય કે જે ઇન્ડિયામાં ઇન્વેન્ટ થઈ હોય અને ઇન્ડિયાએ બહાર આપી હોય. Just think about that. કોઈની જોડે તમારું કંઈક વાતચીત થઈ હોય એમાં તમને ક્યાંક ખબર પડી હોય. There is only one drug that is invented into the India, and that is invented by the Zydus Cadila, Zydus Healthcare Limited, and that is the Lipaglyn, and it is the first glitazar, and the name of that drug is the saroglitazar. Saroglitazar is indigenously invented into the India. There is another one drug that is umbralisib, that is invented by the Alembic, but not into the India. There is a one Swiss partner, Raisin Pharmaceuticals. They partner, they both invent that particular drug, and whole thing is carried out in the Switzerland. So India basically only gifted the one drug. So you can see how the drug discovery is going on. That is only the saroglitazar. Okay. So after all these things, we have some positive statistics for the drug discovery also. We have some positive statistics. Positive for the pharmaceutical R&D worldwide. You can see with all these numbers, we also have average figures for the pharma industries. You can see over here. Okay. So the pharma field are on the top since last 18 years. You can see 2005, and then we are on the top in the R&Ds. Then the semiconductors and other softwares. The most revenue is generated only through the pharma research activities. Okay, so that's why the pharmaceutical companies are invest for the R&Ds. Okay, you can see. There is another some global drug discovery market from 2022 to 2023. Most of the research and most of the things which is drug discovery is carried out into the Asia Pacific. So basically in our region. So very high market over here. And you can see that in this particular year, there is a high market of the high throughput screening, means small molecule synthesis, and the nanotechnology. And the coming market in this Asia Pacific, the Pacific is biochips and the bioinformatics. So we have some good statistics also. And you can see the top companies, top pharma companies in the world over here. This is the bird view of India and pharmaceutical field. This is satisfying something. Okay, so nearly more than, you can see this particular bar chart. In 2024, nearly 5,000 crore today is, we can say, is the Indian pharmaceutical market, and it reaches up to the 11,000 crore INR in 2030. So that is positive thing for us. Within 7 years, it will increase. You can see the data from the clinical trial market. India is the biggest hub for the clinical trial market, and most of the clinical trial 3 is governed into the Indian hospitals. You can see over here. Another thing is the API market. So something about in 2023, you can see the figure of 2023, about 14,000 crores of we can say the market value for the API into the India, and also we covered the China market also this time. This should be increased as per this particular statistics within 3 years about we can say the 2,000 crore market of the API. And this is the export. 90% of Indian export, a pharma export, is the formulation and the API. So these are the some good things that gives us some raise to learn about the drug discovery. These are the top pharma companies. The top pharma companies in India: Sun, Torrent, Lupin, Glenmark. And I'm fortunate that I was the part of the Torrent Research Center in year 2016 to 2018 for 2 years. And I was the victim for that these two molecules. You can see Torrent, that is a new chemical entities, and these both are the disease condition. And the first NC is in phase 2 clinical trial, and the another NC is in the late preclinical trials. So there is a news that this phase 2 clinical trial is fail. Torrent fail in this phase 2 clinical trial, and the drug code is P28. Okay, so but yes, there are some hopes for this particular second NCE, and I'm fortunate that I'm the victim and the part of the at the time when this particular research is going on. Now, this is the general idea about the drug discovery. અહીંયા આગળ તમને એવી ખબર પડશે કે ખરેખર drug discovery માં શું થાય છે basically. Okay, so very first thing is the pre-discovery. That is the gathering much information as possible about the disease and try to understand the nature of that particular disease. So we need to choose the disease first. આવી આપણને ક્યાંથી ખબર પડે? તો તમે જ્યારે કોઈ પણ કંપનીની વેબસાઇટ ઉપર જાઓ તો વેબસાઇટ ઉપર there is one option that is called as a pipeline. When you click on the pipeline, there is a recent research what is going on in that particular R&D. So એમાંથી તમને ખબર પડે કે અત્યારે એ particular company કઈ disease ઉપર કામ કરી રહી છે. Okay, so that is the pre-discovery. You need to identify the disease condition. Another thing is the target identification. Target identification, basically you target the receptor or the enzyme. You know about the receptor and the enzyme. So we need to block the receptor to we can say to send the signals to the to other we can say the molecules. So we need to target the molecules in the body, and targets are the proteins. Then there is a target validation. Target validation is the test that target and confirm its role in the disease, whether that target what we choose it exactly working in that disease condition or not. That is the target validation. Then the drug discovery, that is the chemistry part basically, and find the promising molecule that is called as a lead compound. આપણે lead compound કહીએ, that could become a drug. એમાંથી ભવિષ્યમાં drug બનશે. Okay, so that is called as a drug discovery. Then early safety test, you need to test that pharmacokinetics, absorption, distribution, we can say the excretion, toxicity. You need to find out all these things, pharmacokinetic parameters for that particular molecules. Then there is a lead optimization. You need to optimize that lead. You need to give some substitutions. You need to cut down some functional groups. You need to add some rings to enhance effect, to enhance the binding of that drug with the receptor. કેમે કરીને આપણે drug ને highly binding કરાવવાની છે receptor જોડે, જેથી કરીને disease condition ને એ રીતે એને રોકી શકે. So that is the lead optimization. Then we come to the development phase. That is the preclinical testing. Preclinical testing, that is basically we can say the animal testing. The testing which is carried out on the rats, mice, and guinea pig and everything, rabbits, dogs. Okay, then after that there is a clinical trial exception, we can say the application. If you are inventing the drugs for the, or we can say some for rare diseases or for the cancer, you cannot trial that drug on the humans, healthy humans. So you need to we can say the fill a form as a clinical trial exception application. If there is no such things, you need to go directly to the clinical phase trial 1. There is a healthy human beings, volunteers. Then the phase 2 clinical trials, that is the test of a small group of patients. Basically patient come at the clinical trial 2. Then the clinical trial 3, there is a large group of patients, and you just need to evaluate the safety and the efficacy of your drug. Okay, then the marketing authorization, you need to take some marketing authorization from the regulatory bodies. જેમ આપણે ઇન્ડિયાની અંદર CDSCO છે, ત્યાં આગળ તમારે marketing authorization લેવું પડે. Then and then you can sell your drug, market your drug in that particular country. Then for marketing, you need some bulk. So there is a bulk manufacturing and there is an ongoing studies that is called as a phase 4. Any other ave pharmacovigilance. So this is whole cycle of the drug discovery. So you can see what is the concept behind the drug receptor binding. હવે આપણે એક મુદ્દાને લઈએ અને જોઈએ કે ખરેખર એમાં આપણે શું કરી શકીએ. So this is how the ligand binds with the receptor and that initiate some reactions. You can see this is ligand, the green is the ligand, okay, and this is the receptor, what is the protein, and these are some messengers that initiate the disease condition, we can say. So this is how first receptor is on, and this is how the ligand binds and it initiates or it triggers the disease. So how we target, how we can say the stop the disease. This is the another animation. This is your ligand that is called as a block. Drug છે. And that is competitive antagonism. You can see your drug binds with the receptor. Now ligands cannot bind. And now because of blocking of the receptor, your messenger cannot initiate the disease. This is how basically our drug works in our body. Okay, this is how, and that particular ligand we need to synthesize as a drug. Okay, so this is basically antagonism. Okay, so where do these compounds come from? આ બધું ક્યાંથી આવશે? બધા compounds ક્યાંથી આપણે ભેગા કરીશું? Okay, so that is from the plants, that is from the bacteria, that is from the genetic engineering, and the synthetic compounds. The compounds are also derived from the fungi, also from the animals. So all these are the organic compounds, because all these compounds are, we can say, they come from the living things. So nearly 5,000 to 10,000 potassium compounds or candidates are considered. Okay, so you need a huge library of compounds. તમને બધા compound ક્યાંથી મળે? So there are some databases. હું તમને અમુક databases ના નામ કહું, like Enemine database, Zinc database, PubMed, PubChem, ChemSpider, Campbell. These all are the databases from which there is a storage of the structures, their characteristics, everything in that databases. You just need to take the structure from the databases. Okay, then all the structures which is there in the databases, they all are previously synthesized and characterized structures. Okay, and you just need to do the high throughput virtual screening on the computer models. આપણે સમજીએ કેવી રીતે કરાય. Okay, so here is the we can say the interesting things that is carried out in the drug discovery. That is the initial phase of drug discovery. That is the first thing, the drug target. You can see this is the enzyme or the protein, and this is the docking site. This is the site. This is the cave in which your drug is bind. Okay, and inhibit that particular enzyme. So a target is the molecule that the drug needs to find and act upon, and often it is the protein molecule or such as the enzyme. Okay, the starting point for the design of a new drug is understanding at the molecular level of the disease to treat. So you need to understand the structure of that protein also. That is the protein data bank. Okay, so all the proteins, all the targets are given in the protein data bank. You just need to download that protein structure, and then you need to modify that. Okay, and that modification done by the drug design. Okay, so there is a modern technologies that allows the chemist to work upon the molecular structure to a target molecule and represent it using the physical or the computer generated models. So there are numbers of, we can say, the softwares is available nowadays. Okay, you can do the pharmacophore, QSAR studies, modeling studies, docking studies. Docking is basically how this is the protein and how your molecule bind with that protein, and it gives the docking score. The more the docking score, more your drug is, we can say, the bind, or it is tightly bind with your particular receptor. So you need to screen these things. Okay, so for that you need in silico tools. In silico tools means computers, the heavy computers, the biggest processors, the servers you need for that. Okay, and there are some companies, they they have this type of softwares. અત્યારે એવી કઈ companies છે? Very first is the Schrödinger. Schrödinger is the giant company in this drug discovery. BioVia Discovery Studio, another company. Autodoc, there is a free tool. Okay. Then the Crescent, there is a paid tool. So these all the companies are well known. They are well known in the drug discovery and software that have the drug design and docking studies. You can do the drug design and docking studies in these softwares. So these softwares are full with the complex algorithms. એમાં એટલા બધા mathematical algorithms એની અંદર fit કરેલા હોય એના પ્રોગ્રામમાં. And that's why the precision, the accuracy, what the result from the software, that is same what you predict. Okay, whatever you predict from the software, it is same what you will get in the biological studies. એવું કહેવાય છે કે if you use the Schrödinger software, there is about 85% of accuracy in the prediction of the Schrödinger software. And do you know what they charge for the 1 year's we can say the academic license? They charge 4 lakh INR, Indian national rupees, for 1 year of subscription of the Schrödinger software. So this is what that is going on into the drug discovery. Okay, now we move to the lead compound. How to identify the one compound, one hero from the whole, we can say the in the crowd. So you need to do some we can say the studies for that. That is a structure based drug design, ligand based drug design. There are some concepts. Okay, so with the target identification, the thousands of compounds are made using the technique that is called as a computational chemistry. Okay, and these are the narrow down to the one compound to be studied further. This is called as a lead compound. That is called as a high throughput virtual screening. That is same as the Lenscard tool. આપણે લેન્સકાર્ટની ઉપર સર્ફ કરતા હોઈએ અને એમાંથી આપણે ફ્રેમ નવી લેવી હોય, સો એ પહેલા આપણે ફોટોગ્રાફ ખેંચે અને એના પછી આપણે ફ્રેમ્સને મૂવ કરીએ કે જે ફ્રેમ આપણને શૂટ થતી હોય એને આપણે ચૂઝ કરીએ. So this is the same tool that is called as a high throughput virtual screening. You have a structure of your protein and there is a thousands of compound which is passing near to that protein, and the compound which is most fit to that particular protein, that gives you a score. Okay, so this is how you just identify what is the lead molecule. કયો compound તમારા પ્રોટીન જોડે બાઇન્ડ થશે એ તમને આવી રીતે ખબર પડે. That is called the high throughput virtual screening. Okay, and then that study is followed by the docking study. Docking study gives you a docking score. Okay, and the docking score, that means a lot for any particular chemist. Okay, so after that, when you find some structures which is effective on your drug target, on your protein, then there is a chance for a lead optimization and the scale up. Lead optimization means you just need to add something, you need to just cut something in from the structure. That is called as a lead optimization. Simple. So the lead compound chemistry, or that chemically is modified to produce the number of other structures which is similar to your compound. So the pharmacophore, the basic structure is same. Okay, and in the peripheral region you just need to add some other we can say the substitutions to make to increase the probability of to become a drug, and you can synthesize that things using the parallel synthesizer. Okay, so there are you can do the things in a one time, one shot. Okay, so not only the substitution you carried out, but also you just supposed to mind that we can say to route of the design of the route of synthesis, which route is better to synthesize this compound, whether that route is cost effective or not. પછી બહુ મોંઘો રૂટ હોય, બહુ મોંઘા કેમિકલ યુઝ થતા હોય તો you cannot utilize. Okay, specific and selective synthesis, પછી એમાંથી impurities બનીને જતી હોય, બાય પ્રોડક્ટ્સ બનતી હોય, so એવું ના ચાલે. You need to synthesize the specific compound. Another thing is the yield driven. It should give a specific compound in a specific amount, that is the yield. Easy availability of the raw material, that is also you supposed to take into mind. It should be short. Okay, you should not go with the long we can say the processes, and it should be feasible at the lab level first. Lab level upar a vastu synthesize thavi જોઈએ, then it will goes to the pilot scale, and then it goes, we can say it will it will transfer to the we can say the manufacturing scale. So all these things, this is a very we can say the lengthy process. It requires so much of patience, so much of trials. You can suffer from so much we can say the of the failures during this lead optimization and the scale up. Then we move, that is the biological evaluation. Now we completed with the chemistry part. We have now the series of lead compounds, okay, the series of compounds, and now we just complete one phase, and now we entered into the second phase, that is the animal study. How our particular compound is behave on the biological system, that is called as a biological evaluation and the screening of the molecules. So the very first thing is the in vitro. In vitro kehta ke we can say the artificial environment. We can say we build, and there is a test tube or the culture media, the cell. Okay, so this is the photograph of the in vitro study. Okay, that is the outside the body. Then another thing is called as the in vivo studies. It means the animal studies inside the body. Okay, that is in the living body, and referring to the test conducted into the animal. Okay, and another thing is the ex vivo. Ex vivo means you just need to isolate some organ part and cultured, and we can say the incubate into the into the temperature, and then testing of the drugs. That is the ex vivo, outside the body. So in this particular phase, screening of almost 100 to 150 compounds of one series. एक series જે chemist બનાવી હોય એમાં at least 100 to 150 150 compounds હોય એવી એને 3 to 4 series બનાવી હોય. So nearly preclinical department need to screen 700 to 800 molecules of that particular series on this biological system. આટલું બધું burden હોય એ લોકો જોડે. They need to screen the molecules on this biological system, and after the result, they choose some molecules for further studies. You can see over here, this is the protocol for the in vitro studies. Okay, so the cell line, or we can say the enzyme assay, that is done for the we can say the in vitro studies. This is basically a inactivated enzyme. You need to incubate this enzyme and convert into the activated enzyme. And this is the cell lines. Okay, so after we can say getting these both the things, you need to screen your compounds on these particular biological things. Okay, and because of some biochemical reactions which is carried out in the enzyme at the and in the cell line, it gives us some observations and the findings in a form of some graphs in the statistics. So based on that results, based on that we can say the graphs and everything, we decide that these are the best screen molecules. Okay, out of that 700 800 molecules, you will get 8 to 10 based molecules out from the in vitro studies. Okay, and that 10 molecules now entered into the in vivo studies, that is the animal studies. Okay, so there is a two concepts when you are going for the animal study. There is a two concepts. Okay, one is the preventive study, another one is the curative study. Okay, so before that you need to take a permission from the committee for the purpose of the control and the supervision of the experiments on the animal. Okay, that is called as a CPCSEA. Okay, that is the we can say the regulatory body that gives us the guidelines how to study on the animals, the animal ethics, everything. So you need to take first the permission from that particular body, and then you need to carry out the we can say the experiments on the animal. Okay, that is the animal ethics. So what basically done into the preventive study? Preventive study ma શું થાય? So in preventive study, first you insert your compound or your molecule into the body of your animal. Okay, પહેલા તમે એને drug આપી દીધી. Then there is a induction of disease, અને એના પછી simultaneously parallelly તમે disease induce કર્યો. It means you add the disease in the body of animal. Okay, and then see what happened in the presence of drug, whether the disease is increasing, it's we can say the it's bombarding or not. Another study is called curative study. Curative study means first in animal, you just insert the disease. Okay, first you just insert the disease, okay, and then give the drug treatment, that is your molecule, which is screened into the that cell trials. Okay, so you just need to administer the drug in a different doses, high, medium, and low. So you can see, you can observe that કે કયો dose effective છે, high dose effective છે, medium low કે low effective છે. Then finally you need to monitor the we can say the physiological parameters and the animal behavior. Okay, and you will get some research conclusions out of that and the new findings for your particular molecule. કે ખરેખર molecule જ્યારે animal ની બોડીમાં જાય છે એના પછી શું behave કરે છે, whether it is binding with that particular enzyme or protein or not, that we can predict using the biological studies, that is the in vivo studies. Okay, so after completion of these things, we entered into the ADMET studies. ADMET studies mean whatever you will get, you need to conclude that out of these 10 molecules, these 2 molecules are the best molecules in the animal studies. So check their ADMET, absorption, distribution, we can say metabolism, excretion in the body. Okay, so basically what is metabolism, what is the absorption? Absorption, it means how will it get into the blood circulation, whether it goes into the blood circulation or not. Okay, so that is the testing called absorption testing. Then the metabolism means how liver react on your particular drug. Okay, so that is the metabolism. Distribution, that is the first stage before the metabolism. It means how your drug is totally distributed into the body, whether it is distributed or not, how they bind with the protein. Okay, that is the thing that is called as a distribution. And final thing is the excretion, how your drug is eliminated from the body. That thing you also need to know. Okay, otherwise it causes some toxicities. So it means you also do some toxicity studies also. Okay, whether your drug is harmful to human body or not. Jo drug harmful hoy, so there are lots of toxicities. Carcinogenicity, genotoxicity, mutagenicity, hepatotoxicity. Okay, the nephrotoxicity, it means failure of kidney. Neurotoxicity, that means it ruptures the neurons. So you need to evaluate all these things, then your chemical moiety go further in other studies. Okay, so that is the another thing. And this is how the lead series, what chemistry people synthesize. So this is how the path of that particular chemicals. Okay, like that series 1. So they may fail because of the we can say the pharmacokinetic problem. Series 2 maybe fail because of the metabolic issues in the human, we can say in ADMET studies. Only the drug candidate that pass in all these tests, it only can reach to the clinical trials. So now we move for the clinical trials. Sorry. So this is the clinical trials. It means now the best molecule that is come to the human volunteers. Okay, so in phase 1 clinical trials, that is 20 to 100 healthy volunteers, and you need to just identify and confirm the safety of your drug in the humans. Okay, that is a very small we can say the group. Okay, and 10 to 20 candidates of drug are there in that particular phase 1 clinical trials, that are the backup molecules. Okay, 10-20 is the backup molecules of your lead structure, and only 5 to 10 are passed in the phase 1 clinical trials. Okay, these are the testing in which there is a we can say the most chances, or we can say the higher chances of your project failure. Okay, so that molecule P28 is failed into the phase 2 clinical trials because of the dose. Okay, so phase 2 clinical trial, it means 100 to 300 patients. Now the drug is administered into the patient now. Okay, and we need to check the effectivity, dose, and the delivery method into that particular patient. If phase 2 clinical trial is passed, then we move for the phase 3 clinical trial, and that is the 1,000 to 5,000 patients, and there is a multiple location. Okay, So in multiple locations, you need to carry out the trial, that is phase 3 clinical trials, and only 1 and 2 drug pass this phase 3 clinical trials. Okay. Sorry. So what we need to observe in phase 3 clinical trials, that is the reconfirmation of findings, whatever we find in the phase 1 and phase 2, whether it is repeating in phase 3, identify the best doses regimen, exactly kyo dose apai, કેટલા time માટે અપાય, so એનું concentration હોવું જોઈએ, that all the things which carried out in the phase 3 clinical trials. And you know, the Gujarat and the India is the hub of clinical trial. આપણે clinical trial નું hub છીએ. Okay, that called clinical research organization. જે clinical trials કરતા હોય એને કહેવાય clinical research organization or contractual research organization. So in Ahmedabad there are many clinical research organizations. બરાબર, ઘણી બધી છે, જેમ કે Epser Life Science, then Vida, then Astron, then Lambda Therapeutics, that is the part of the Intas, then the Clientha. These all are the clinical research organizations. They monitor the clinical trials basically. Okay, so you can tell that this is the phase in which there is a maximum failure. આ જગ્યા પર failure આવવાના બહુ chance છે. There is a one brother of metformin. Its name is fenformin. Okay, so fenformin is failed in the clinical trial because of the severe acidosis. Two other molecules, that is betamestate and merimestate. This betamestate and merimestate also failed in phase 3 clinical trials. These two drugs are utilized for the rheumatoid arthritis. વિચારો, બરાબર, સંધિવા માટે આ drug use થવાની હતી, but they fail. What is the side effect? You know, the side effect is severe mucoskeletal syndrome. When you take this drug, there is a severe pain in the joints. You can't tolerate. Okay, so that's why these drugs are failed in phase 3 clinical trials. So how they basically do the clinical trials? So there is a group 1, they provides the normal things, okay, and group 2 they provides the pain relief cream. This is the example. Okay, in normal group there is no active pharmaceutical ingredient, એમાં કોઈ drug ના હોય. Okay, and they both the groups now exposed to the pain. Okay, so we can easily predict the group 2 feels very less pain because they applied that particular pain relief cream. So this is how the clinical trials is governed. Okay, but before that you need to fill up this form. Not we, but what the volunteer is. Those who involved in the clinical trials, they need to fill up this particular form. That is the informed consent. Okay, so informed consent form, they need to fill that I confirm that my participation is the volunteer. Okay, voluntary મારી જાતે આની અંદર participate કરું છું. I understand the possible risk and attend to the experiment. મને બધી જ વસ્તુ ખબર છે કે આ ડ્રગની સાઇડ ઇફેક્ટ શું છે, એનાથી કયા કયા રિસ્ક છે હું જાણું છું. And the third thing is I agree that my data may be shared with the other for their analysis. Okay, so you cannot hide that particular data. You need to tell that data and you need to transfer that data for the other purposes. So this is the consent form that is filled by the volunteers before going into the clinical trials. Okay, so there is another mechanism that is called as a placebo effect. You need to identify the placebo effect in the clinical trials. How? Why need to we can say go for the placebo effect? Because of mainly checking for the central nervous related drugs, okay, or psychiatric drugs. Okay, so in this particular clinical trial you need to identify the effect of drug. Okay, and you need to tell that the effect is only because of drug, not from the inactive ingredients. Okay, so if you give the blank placebo to the patient and if he or she feels good after that, okay, so you need to identify that, okay, whether the drug caused the good effect or whether the we can say the emotions or the psychiatric, we can say the things that cause the effect. So you need to give the placebo therapy in the clinical trials also. So now we are towards the end. Okay, drug plus the clinical trials, then what is next? પછી શું કરવાનું? જો drug pass થઈ જાય clinical trials, so then you go for a licensing. તમારે એના license લેવું પડે. Okay, so the pharmaceutical company must apply to appropriate authorities to produce the market of that particular drug. So you need marketing approval. Okay, so here is some agencies like CDSCO, US FDA, national, we can say the health service. These all are the we can say the regulatory bodies of the different countries that regulate the licensing or marketing of that particular drug. Okay, what is other thing is you can say the taste to taste the use in the combination with the other drugs or the patient with the different conditions. Okay, so that is called as a phase 4 clinical trials, and that particular trial is totally governed by the pharmacovigilance team. Pharmaco PV team કહીએ આપણે એને, એના વડે આખું મોનિટર થતું હોય. Okay, so adverse drug reaction also, they report the adverse drug reaction in the phase 4 clinical trials and everything. Okay, now this is the chart that indicates when to file. Okay, when to file. So regulatory filing during the drug development process, so you need to file this IND, that is the investigational new drug application, at exactly after the preclinical studies. તમે જેવી એનિમલ સ્ટડીઝ પૂરી કરો એના પછી તમારે ક્લિનિકલ સ્ટડીઝમાં મૂવ થવું હોય, you need to file IND. Okay, so that is the process. After completion of your clinical studies in phase 3, you need to file NDA, new drug application. Okay, so new drug application is filing at the phase 3 clinical trial. Okay, so this is the chart. If you fail in clinical trial 1, you need to further start from the clinical trials. Okay, if you fail in the trial 3, you need to go to the trial 2. If you fail in trial 2, you need to go into trial 1. Okay, if the FDA reviews and approve, okay, then you will get the marketing rights. And if you fail to submit some results and you not pass, you are terminated. This is the failure. Okay, so this is what is the filing. Now the most important thing is the patenting. Okay, patenting, it means intellectual property rights. You need to register your idea, and no one can follow, no one can infringe your particular innovation. So this is important for a drug discovery. It is a long process. There are many fails into that. Okay, and pharmaceutical industries invest hundreds and billions of pounds in the patenting. Okay, why to patent? Because you need to recoup the investment. તમારી investment માંથી પાછું કંઈક મેળવવું છે. Okay, after patenting, you have exclusive rights for only with you. You need to invest more in the other R&Ds. બરાબર છે, તમને જો પૈસા આવશે, તમે જે ગેઇન કરશો એમાંથી તમે બીજા પ્રોજેક્ટ્સની અંદર ઇન્વેસ્ટ કરી શકશો and make a profit. That's why there is a patent. I need to tell you that there is a IPR department, intellectual property right department in any particular pharmaceutical company. Do you know they are in a high alert? They are always in a high alert. They always do the literature. They always literature karta hoy. Okay, they always cross verify the things. Whatever the scientists do in the lab, they need to cross verify with all other patents whether we are infringing the patent of other or not. આપણે કોઈ બીજાને તો infringe નથી કરતા ને. Okay, so they are always in alert. Okay, they always assist the scientists for their methods. તમે જે મેથડ યુઝ કરી રહ્યા છો લેબની અંદર ડિસ્કવરી માટે, તો તમારે ફોલો કરવી જોઈએ કે ના કરવી જોઈએ. They always assist you. The ingredient and the process, they always we can say they monitor the ingredients and the process what you are doing. They have lots of database access. એ લોકો જોડે બધા database નો access હોય. Okay, એની જોડે SciFinder, SciHub, બીજા ઘણા બધા છે, એ બધા જ એ લોકો જોડે access હોય. That's why they can identify, they can find the patents whether it is infringing or not. Okay, they work day and night. Okay, the IPR department is so much important in the pharmaceutical industry. They are the legal department, and it is a white collar job. Okay, but there is another thing. Okay, as I'm working in I have some experience in working with this research group. The data is very confidential. The work is very confidential. You cannot tell even your colleague. બરાબર છે, બહુ confidential વસ્તુ હોય. The data and the work is highly confidential. The internal data storage is there in the company and the R&D. લોકો જોડે પોતાનો એક hard drive નો data storage drive હોય. એ લોકો ક્યારે R&D ઉપર data store ના કરે. They store the data on the other location. If there is a fire in the R&D, what happened? The data also burn. So they store the data on the other side. Okay, this is the real things in the R&D. હું તમને એ કહું છું. Okay, there is no one know the final product structure other than the VP. કોઈને ખબર ના હોય કયું chemical structure અત્યારે screen થઈ રહ્યું છે, કયું અત્યારે, not even the cell, we can say the cellular scientist or the preclinical scientist, they know the structure of what they screen in the cell line. Only the vice president and the general manager, they know the structure of the chemical. Okay, you cannot converse with your colleagues and not ask the questions to your boss. તમે કોઈ ક્વેશ્ચન ના પૂછી શકો એને કે આ કયું સ્ટ્રક્ચર છે, ક્યાંથી આવ્યું, કયા ગ્રુપ છે, કઈ ના પૂછી શકો. Okay, another thing is there is a rotation in work. You are not we can say at the same work for a longer period of time. તમારું always rotation થાય. તમે અત્યારે આ વસ્તુ synthesize કરી, within a 2-3 weeks you are going to the another series. So you cannot remember what you done previously. Okay, so that is the thing. There is an empty hand job. તમારે કશું લઈને નથી આવવાનું, કશું લઈને નથી જવાનું. That is the restrictions. That is an empty hand job. Full checking at the company side. કંપનીમાં તમે એન્ટર થાવ ત્યારે તમારું ચેકિંગ થાય. કંપનીમાંથી તમે બહાર નીકળો ત્યારે તમારું full checking થાય. There is a data security devices. ત્યાં આગળ data security devices હોય. Okay, there is a blind testing. There is a secret coding of the compound. દરેક compound એક secret code આપેલો હોય. Do you don't know કે કોડની અંદર કયું compound છે? Okay, you don't know. Okay, there is a secret coding and the experiments, and there is restricted entries in R&D. ક્યારે તમને R&D visit કરવા નહીં દે. કોઈ પણ સ્ટુડન્ટ્સ કે કોઈ પણ બીજાને બહારેથી you can't visit R&D. તમને R&D બહુ ખાસ visit નહીં કરવા દે. There is always 24/7 CCTV surveillance. Okay, and the last thing, one mistake and you fired. One mistake, only one mistake and you fired from that particular R&D, from that company. So this is the consequences that is related to the drug discovery. Okay, and this is the second last slide, I think, and how basically the company makes the money. Company કેવી રીતે પૈસા બનાવે. Okay, so you can see at the phase 2 they basically filed a patent. આ જગ્યા પર એ લોકો patent file કરે. Basically this is the file of patent. After 4-5 years the patent is granted. Okay, so there is a granting of patent. At that time the study is thus parallel to the phase 3 clinical trials. જ્યારે phase 3 clinical trials ચાલતા હોય ત્યારે exactly patent grant થાય. Okay, and after the product approval there is a long process of product approval. So there is a product approval, and then after 12 years of patent, we can say the filing and everything, and after you can say after 7 to 8 years of granting of your patent, company that we can say they get the data exclusivity rights. So these 5 years are the best years for the company. આ જે જગ્યા છે ત્યાં આગળ company કમાઈ લે. Okay, so this is how company makes the money in this particular period. And after the patent expiration, generic drug આવી જાય. Generic market, જે API market અત્યારે ઇન્ડિયાનું ચાલે છે, that is the generic market that come into the market. Okay, so what are the new updates? This is the last slide. What is the new updates in drug discovery? There is an involvement of the AI now, artificial intelligence, in the drug discovery. Okay, for the predictions of the new compound. What is new? There is a flow chemistry. What is a flow chemistry? You need to feed one side, you will get the output continuously. So that is called as a continuous manufacturing process. There is a no reactor. There is a no conventional way to synthesize the compound. Okay, there is a continuous process. You just need to feed the things over here and you will get the output over here. Okay, continuous output. Okay, what is the another thing? That is the green process and the hazard free process. Okay, what is a new thing in the drug discovery? You need to carry out the synthesis in a hazard free environment. You need to follow the pollution guidelines, and you need to also concern about the water use. Another thing is the robotics in the drug discovery. Okay, that is the targeted delivery, and the robo drugs are used for killing the cancer. Okay, So that is called as a robotic drug. Another main important thing is the impurity profiling. This is the main cause of the pharma shutdown. Okay, nitrosamine impurities, genotoxic impurities. So you after completion of the drug discovery for product life cycle management, you need to carry out the impurity profiling periodically. After 1 month, after 6 months, koi navi impurity generate nahi thai gayi na. Okay, that is for the impurity profiling. Then drug repurposing. Use the one drug for multiple, we can say, the treatment. That is called a drug repurposing. Then the animal ethics. You need to just we can say that is the thing that you need to just refine your thoughts about the animal ethics. Okay, so to minimize the use of the animals, to rehabilitate that animals, to do the other studies. એક વખત તમારી સ્ટડી પતી જાય એનિમલ પર, પછી એને એક વોશિંગ પીરિયડ આપવામાં આવે, એના પછી એના એ જ એનિમલનો ફરી વખત યુઝ કરવામાં આવે. Okay, so that is the new thing or in the drug discovery studies. Another thing is the biosimilars. Okay, that is we can say in the R&D, the biosimilars and related things are growing fast. The Biocon is the company that makes the biosimilar. And finally, that is the 3D drug printing technology. Okay, this is the new thing in the drug discovery. That is the 3D printing of drug to change its dosage form and frequency, sustained release, then the immediate release. You need to just print that drugs in the printer. So there is a new technologies in the drug discoveries. So this is the end of my presentation, and this is the summary. Drug discovery, phase 0, phase 1. This is clinical trials, and then you need to go for a licensing, and finally there is a pharmacovigilance. That is the phase 4 clinical trials. And I want to just dedicate this talk to all the researchers who are involved and working for the drug discovery, the animals who sacrificed their lives for our betterment. So I dedicate this talk to all that animals, and also the human volunteers, who involved in the clinical trials. So thank you very much. Thank you very much, guys, and for your patient listening and your involvement. I would like to thank Gujarat State Pharmacy Council for providing us a platform to discuss such things amongst you. Thank you, Induka Kaikwala College of Pharmacy and the Charutar Vidya Mandal, the CVM University, for inviting me to deliver this talk on the drug discovery. I need to special thank to Dr. Harsha ma'am, Dr. Dimal sir, Dr. Usman sir for their support and lots of love. And I would like to thank also to my institute, Ramanbhai Patel College of Pharmacy, and my university, Charutar University of Science and Technology. So thank you very much, guys. Thank you.
Thank you, sir, for this wonderful and knowledgeable session. Sir, if participants have any question, they can drop their questions in the question answer session or the chat box.
Okay.
Till now, there is no questions from the participants.
Okay. So if you have any question or doubts, you can contact me. I already said that my contact details over here. So you can just take a screenshot. Okay. And you can contact me. You can mail me. Okay. Anytime. Okay. So that's the thing. Okay. Thank you very much all. Okay.
It is indeed a pleasure to share that Nikunj sir is also our alumni of IICP, and after completing his B.Pharm, he did his M.Pharm from MS University in medicinal chemistry department. So again, it is a proud moment for all of us who are present here. So thank you very much, Nikunj sir, for sharing your valuable thoughts. One thing I would like to share with the participant that when you are filling your attendance link, there is IICP registration number which is written, where you have to write your IICP 001 or 002, whatever it may be, your number is. Many participants are writing their GSPC registration number, G number લોકો લખે છે, એટલે આ વસ્તુ ખાસ ધ્યાન રાખજો કે તમારે IICP નો registration number IICP માં લખવાનો છે, IICP 001 કે જે તમારો નંબર હોય, બરોબર. અને જ્યાં જીએસપીસી નો રજિસ્ટ્રેશન નંબર માંગ્યો છે ત્યાં જીએસપીસી નો નંબર લખો. ઘણા બધા લોકોએ આ ભૂલ કરેલી છે ગઈકાલની લિંકમાં. સો એટલીસ્ટ ટેક કેર કે એ વસ્તુ હવે ઓનવર્ડ્સ ના થાય. બરોબર છે? ઓકે, થેન્ક યુ.
Moving towards our last plenary session for today's online refresher course. Take the attitude of a student. Never be too big to ask questions. Never know too much to learn something new. Now, the last session will be conducted by Dr. Ashok Mahajan sir, Professor Indukaka Epkola College of Pharmacy, New Vallabhyanagar. He would be delivering a talk on concept of generic drugs and its selling in India and abroad. I would request Dr. Veena S. Patel ma'am, Associate Professor, Department of Pharmacognosy, Indukaka Epkola College of Pharmacy, to introduce Dr. Mahajan sir.
Thank you, madam. Very good morning to all participants in last session of 2 days online refresher course organized by Indukaka Epkola College of Pharmacy, which is sponsored by Gujarat State Pharmacy Council. It's my immense pleasure to introduce our speaker for the session, Dr. Ashok Mahajan. Sir did his B.Pharm from MGVM College of Pharmacy, Nasik, Pune University, M.Pharm from KLE College of Pharmacy, Belgaum, Rajiv Gandhi University of Health Science, Bangalore, and PhD from Ganpat University, Kherwa, Gujarat. Sir is having academic experience of 17 years. Currently, sir is working as a professor and head at Department of Pharmaceutics, Indukaka Epkola College of Pharmacy, New Vallabhyanagar, a constituent college of CVMU University. During his academic career, Dr. Ashok and his team received third prize for Best Research Project in IICTE MHRD organized COVID-19 Mega Challenge for Fighting Against Corona in April 2020. Sir ranked first position during a Tech Pitch event in GTU Tech Fest 2019 held at BEATS Education Campus. Dr. Ashok is the recipient of Best Performer Award at APP 5th Indo-Malaysian Conference. Sir fetched a research grant for minor research project of ₹2 lakh from Bhuj Post. Sir received grant under different units of SSIPs like GTU SSIP, Student Open Innovation Challenge SSIP, and Government of Gujarat SSIP and iHub, worth amount rupees 80,000, 90,000 and 1,75,000 respectively. On his credit, 2 granted Australian innovation patents are there. His 2 Indian patent has been published, and Sir has applied for 3 more Indian patents. Sir has published 29 research articles in national and international journal of repute. Sir has written 2 book chapters in Springer Publisher and also published 1 book. Dr. Ashok is a reviewer in various national and international journals. With this brief introduction, I invite Dr. Ashok Mahajan on behalf of Indukaka Epkola College of Pharmacy to deliver a talk on concept of generic drugs and its selling in India and abroad. Welcome you, sir.
Yes, sir.
Thank you.
Sir, your voice is not audible. Yes, now it is audible.
Sir, I think it is not visible. Please again start it so it will be easy. Sir, your voice is not audible. No, sir. No.
Now it is audible?
Yes.
Okay. Sorry for the introduction. Can I start?
Yes, yes.
Okay, okay. Thank you for inviting me to deliver a talk or share my views on concept of generic drugs and its sale in India and abroad. You all know that generics nowadays are being replaced by the branded drug. So in my presentation, I'll be explaining you about what these generic drugs are all about. Then also I'll be talking about the branded drugs. In addition to that, how generic is being practiced in India and how generics is being practiced in USA. So I'll just have a comparison and how the pharmacists are substituting or giving a generic drug for a branded drug molecule. So before starting with the generics, I would like to brief you about what are branded drugs. So these branded drugs are also called as innovator drugs. For example, if a Sun Pharmaceutical is there and they want to develop a drug for or a formulation for a lung cancer, and that formulation for a lung cancer is not available in India or it is not available worldwide. So they are the pioneers who has developed a formulation and they have put that formulation into the market. So once the formulation comes for a first time in the market, so that formulation is referred to as a branded drug. So whenever this formulation is being developed by the Sun Pharmaceuticals, they has to go for various preclinical and clinical tests so that they assure that whatever the product goes to the market, it is safe and it is effective and it claims to treat safe for the human use. So during this branded drug product journey, generally, as if you all know that whenever first time whenever the drug product comes to the market, it takes a long journey of near about 10 to 15 years. So first stage talks about the invention, the discovery of the molecule. Then it goes with the animal studies. Once the animal studies are through, and if in animals the activity is shown in a promising way, then they go for clinical trials. And Mr. Nikunj Parekh sir has explained in detail about the details of the clinical trials. So generally clinical trials talks about the studies on the human beings where you are proving your drug molecule, it is safe and effective. Now, once the clinical trials are over, and if the drug molecule passes successfully all the clinical trials, then company has a molecule which will be put into the market. So before that drug molecule is being put into the market, company always go for a patent. So because, as I've said that, I've given the example of a lung cancer formulation, which is been developed by Sun Pharmaceuticals. So whenever Sun Pharmaceuticals has a molecule which has passed
Ashok
all,
sir,
huh?
sorry to interrupt you, but slide show is not visible and the slide is not moving. Give us some few minutes to resolve the problem.
Okay, fine.
Okay.
Now slides are visible?
Yes, it's completely visible, sir.
Slides changing when I'm changing the slides?
Yes, sir, it is
Yes,
changing
yes.
when you are shifting the slides.
Yes, sir, changing.
Okay, fine. Okay,
Yeah.
fine. So what I was telling you, that whenever Sun Pharmaceutical is a pioneer for that lung cancer drug delivery or drug formulation, it will go for a patent. Now that patent claims that, or depending upon the region where the patent has been filed, it is generally having expiry date of 20 years. So Sun Pharmaceutical is the first pioneer who has put that molecule for a lung cancer in the market, and that Sun Pharmaceuticals will enjoy the rights for manufacturing and selling for the entire 20 years. None of the company, because it is a patented molecule. So this patented molecule, as I've told you, it is also referred to as a branded molecule. It is also referred to as a innovator's molecule. Now, when this patent gets expired, or whenever it is at the age of the expiry, then the competitor pharmaceuticals will start working on the generic molecule, and then that molecule will be put up into the market. So this is the cycle of innovation and competition. Branded company means, I have given you the example that Sun Pharmaceuticals have invented a molecule. So once they have invented a molecule, it is a new molecule. So they have a monopoly in terms of patent. So they enjoy the rights exclusive for marketing and selling that drug molecule for 20 years. So after patent expiration, then there is an open market for generic competition. So at that time, one point I would like to tell you that once the patent is there, during these 20 years, the company doesn't earn a single amount from all those studies. So once a patent is been granted for any pharmaceutical company, in order to recover the losses for these, or in order to recover the losses which has been put by that company during these 15 years, that is being recovered with the help of the MRP of the tablet. So generally we see that whenever any patented product is there or any branded product is there, the cost is very high. So whenever the patent gets expired, then generic companies come into the market and generics are distributed. So the branded company loses the revenue. So once a generic companies put the molecule, then the competition is there between the branded and the generics, and the cost then reduces. So this is the cycle of innovation and competition. So whenever you talk about these generic drugs is nothing but a copy of an original branded drug product. Once the patent of the original product is expired, the other pharmaceutical companies are able to create the own version of the medicine. Own version means they can't copy. The excipient should not be same. The excipient differs from the original or branded drug product. So generic drugs is chemically equivalent. Chemically means, for example, if I want to give example of branded and a generic, chemically means it will have the same API. API means it will have the same drug. For example, if I say a branded tablet of paracetamol, means it will contain paracetamol, and a generic version of paracetamol, it will contain a paracetamol. So that is, I'm talking about a chemically equivalent. So it has a lower cost of branded, lower cost as compared to the branded name. Now, whenever any pharmaceuticals want to develop a generic version of a generic version, there are certain requirements. Those requirements are generic drug should be having a same active ingredient. For example, if there are two generic products, then both of the, if there is one generic product and one branded product, in both generic and in branded drug product, the API should be same. For example, if you have a paracetamol tablet, the generic should contain paracetamol and the branded product should contain paracetamol. It should not happen that the generic will have a paracetamol along with diclofenac. So it is not a generic. So first is you have to have a same active ingredient. Second one is it should having the same dosage form. Dosage form means if a branded is a tablet, then generic should be a tablet. If a branded is a syrup and if a generic is a tablet, it should not happen. So it should have same dosage form. Third one is it should have same strength. Strength means the dose. For example, if I'm talking about a branded version and it contains 500 mg of paracetamol, so generic should also contain 500 mg of paracetamol. It should not happen that generic should have 250 mg. So these are one of the basic requirements when any pharmaceutical company wants to develop a generic drug molecule. The next is whenever the therapeutic action should be same. Means, for example, now I have given you the example of paracetamol. It is used to decrease the body fever. So the both of the tablets should have the same pharmacological action into the body. Then next one is the inactive ingredients. Means apart from the drug, a lot of excipients are being used in the formulation of that dosage form. So these excipients will not affect any or it will not give any kind of an effect into the body. So it says that inactive ingredient should be safe and it is not hindering the absorption, distribution or ADME characteristics of the drug molecule. So these are some of the basic requirements for generic drugs. Now, what is allowed in the generic drugs? Means what differences can be there between a branded drug and a generic drug. So these are some of the differences which are being allowed. For example, a branded drug and a generic drug may have different salt. For example, I'm talking about a diclofenac and diclofenac sodium. So if an original drug product is having a diclofenac and if a generic version goes for a salt form, got it? It is being allowed as per the regulations, provided the salt and the excipients does not affect the safety and efficacy of the drug molecule. I am again repeating, the salt version is only allowed whenever the salt form does not significantly affect the efficacy. Efficacy means it does not affect the or it does not decrease any kind of an action into the body. So in that case, and this salt is been allowed by the various regulatory bodies, USFDA regulatory bodies, European regulatory bodies, as well as Indian regulatory bodies. So this is the first difference. It can have different salt. Second difference is it can have different excipients. Color may be different, flavor may be different, preservatives may be different. Then it may have a shape. The original branded product may have a or it is having a circular tablet. The generic may have a different shape. It may have a different size. It may have different scoring also. And the important point is also that the drug expiry date will be also different than the generic as well as compared to the branded drug product. The manufacturing process also can be different, and they may have a different product name, of course, and also packaging. So these differences are allowed between a generic and a branded drug molecule. So all these companies, whenever they develop these generic formulations, they keep these things into the mind. Now, the next question comes that whenever you substitute any generic version for a branded molecule, the question always comes to the one's mind, whether it is a patient, whether it is a pharmacist, or whether it is a doctor's, how these generics are, or whether they are safe or whether they are effective. So in order to prove the generics are effective and safe, all the pharmaceutical companies has to do the bioequivalence studies. Now, what do you mean by bioequivalence studies? Bio means biological system. Equivalence means companies, these bioequivalence studies, equivalence means same. This means that whenever you are giving a branded drug product to the human being, and whenever you are giving a generic drug product to the human beings, it should show same effect. So that is referred to as bioequivalent studies. Now, whenever I'm talking about the different regulatory bodies, because US FDA is considered to be one of the most stringent regulatory bodies for any kind of a drug approval. So if you talk about US FDA or if you talk about India, they both countries follow same guidelines that whenever a generic is being compared with the branded drug molecule, they have to perform the bioequivalence studies. So what is bioequivalence studies? The bioequivalence studies is nothing but it is a rate and extent to which active ingredient becomes available at the site of the action. In short, we have to prove, or the pharmaceutical company who is manufacturing the generic version, he has to, or that company has to prove that generic is showing the same effect in the human beings. So if it is showing the same effect in the human beings, then and then only that generic version can be substituted for the branded drug molecule. Got it? Now, during these bioequivalence studies, they will compare basically certain parameters. So those parameters are Cmax, AUC. Now, what is Cmax? Cmax is nothing but whenever I'll be giving a branded drug molecule, what is the maximum concentration of that branded drug which goes to the circulation, or which goes to the systemic circulation. Similarly, I'll be checking the AUC. AUC is nothing but, in simpler words, it talks about how much molecules of drugs are going into the systemic circulation. For example, I am taking a paracetamol tablet, and paracetamol tablet contains 500 mg of molecules. So during absorption, it may possible that all the 500 molecules will not reach to the systemic circulation. So out of 500 molecules, maybe 400 molecules which reaches the systemic circulation. So that is referred to as AUC. So during these bioequivalence studies, we are checking that whether the Cmax of the generic medicine and whether the Cmax of the original medicine is same or not. Similarly, we are checking the AUC of the generic medicine and AUC of the original brand or the innovator medicine is same or not. If it is not same, then it is not bioequivalent. If it is same, then and then it is considered to be bioequivalent, and then we can substitute that drug molecule as a generic version. Now, there is a lot of confusion about the myths or the facts about generic drugs. Generic drugs are not inferior in quality to the branded drugs. So I just wanted to say that the generic drugs are equivalent to the branded drug molecules. They are not inferior in quality. Then generic pills do not contain less active ingredient than the branded drug products. No, it is not so. As I've already given you the example that if paracetamol, one of the product of paracetamol is branded, it will have 500 mg dose. The generic will also has 500 mg dose. Got it? So if I talk about now, this 500 mg dose is for adults. Now, if you are preparing a generic version for smaller children, the dose is 125. So in that case, the branded drug should have 125 mg and the generic version should have 125 mg. So it goes in that way. It doesn't happen that active ingredient is less in generic or in case of the branded drug product. It is not so. Now, the third point is that inactive ingredients in generic do not affect the absorption. Got it? So it is not so. So if all the excipients which are being used in the generic, it has been also seen that they will not affect any kind of an absorption during, so that ultimately the absorption is responsible for the action into the body. And patients who are well controlled on a branded medication can be switched to a generic medication. So a lot of confusion is there, because if for a period of 5 years, if a patient is giving or patient is taking a branded medication and the cost of that medication is too high for him, then we can substitute a generic version for that patient. It is not going to happen that if the patient is shifting from a branded to generic, it will not show any kind of an differences. It will have the same effect what the branded medication is giving. Whenever you talk about these generic drugs in generic drug, it's sale in India and abroad, there are different situations. Got it? So in Indian scenario, If anyone wants to put a generic, for example, if a patient is there in India and that patient goes to the pharmacist, got it? So in India, there is no as such any kind of an rules or guidelines that this product should be substituted against that drug molecule. Got it? So pharmacist will push those brands that offer higher margins. But if you compare the same things in USA, the scenario is different. The pharmacist can legally substitute generic medicines for branded drug product which are mentioned in the Orange Book. Now, what is Orange Book? Orange Book is nothing but one kind of an official book which is being published by US FDA. And this book contains all the list of patented product as well as the generic product. And hence, when in USA, if a patient goes at the chemist and he or she wants to have a generic version, so at that time the pharmacist needs to see what basically the substitution can be possible for that drug molecule. So I'll just be explaining you in the coming slide how the substitution takes place in the USA in the for any kind of a branded drug product. Now, what is Orange Book? Orange Book, the name of first of all, why the name Orange Book? It is nothing related to any kind of an oranges. The first book, which was being published by US FDA in October 1980, the color, the cover page of that book was orange. Hence, the name is called as Orange Book. Got it? Now, this Orange Book basically contains approved drugs, products with therapeutic equivalence and evaluation. Now, the latest version, I'm talking about 2023. So it is the 43rd edition. So every year, this Orange Book is being published. It is updated on a monthly basis. So what all generic products which are coming to the market, what are patented products which are coming into the market, also what products which are being removed from the market. So all the information is been there into this Orange Book. Now, generally, if you talk about this Orange Book, what this Orange Book will give you an idea. One important thing which I want to tell you is that in USA, it is mandatory for all the pharmacists that you have to keep these Orange Book in the medical shop, right? Also, this Orange Book is also kept in the hospital. So a doctor or a nurse or a pharmacist who wants to go for a substitution for a branded drug molecule or generic version for a branded drug molecule, he has to refer this Orange Book. Now, this Orange Book has got different sections. It starts from introduction, how to use, the drug product list, the patent information, and the exclusivity information. From the point of a community pharmacist, the most relevant section is drug product list. Now, this drug product list is nothing but it gives an idea about all the formulations available in the market, whether it is a generic version or whether it is a branded version. So these drug product list in the Orange Books will give you an idea about the branded drug products. Now, these branded drug products in the Orange Book are also referred to as reference listed drug. It is called as RLD. Now, whenever any company wants to do a bioequivalence studies, means bioequivalence studies, which I have told you that comparison of generic with the branded drug product. So at that time, pharmaceutical companies has to choose a reference listed drug from this Orange Book, and they will be comparing their generic version with the branded drug product. So during the bioequivalence studies, the pharmaceutical companies has to use this RLD. RLD means it is nothing but the branded product from Orange Book to compare its generic version, and it is mandatory everywhere. Got it? Now, there are different codes which are being given in the Orange Book because of all many products are being listed into the Orange Book. So how one will able to know that whether a generic version is available for that drug product or not? So in that, for that purpose, TE code is given. T stands for therapeutic, E stands for equivalent. T means that is nothing but the pharmacological action which has been shown into the body. So that is referred to as therapeutic equivalence. So therapeutic codes or TE codes are being given for each drug product in the Orange Book. Now, a TE code is always a 2-letter code, and sometimes it is being followed by a number. For example, always the code is there in form of a 2 letters. Got it? AT or it is BT. If the first letter is A, then the A code means that it is therapeutic equivalence. Got it? And the second letter of the code, it talks about the dosage form. For example, if I'm having A first letter, it means therapeutic equivalence. This therapeutic equivalence means it talks about that a generic version is available for that drug molecule, and T stands for dosage form. So T is referred to as topical product. So in some drug products, BT is written down. So if a B is being a code, and that is a first letter code, so it talks about that there is no therapeutic equivalence for that drug molecule, means a generic version is not available for that drug molecule, and T stands for topical drug product. So I'll just tell you. So these are the different codes which are being given in the Orange Book. So these are all the A series, which talks about there are generic version available for all these drug products. The second letter talks about nothing but the dosage form. So A stands for oral solution, N stands for aerolized drugs, O stands for oily solutions, P stands for injectable aqueous solution, T stands for topical products, which I have given you the example. Now there is also one classification which I have told you, that is BC/BD. So B stands for these drug molecules bioequivalent is not available. So depending upon the dosage form, every drug molecule is being coded, and this coding is being done based on the therapeutic equivalence. Now I've just taken one example from the Orange Book. Now I just wanted to give an example of erythromycin. This is the data which has been given in the Orange Book. So this is the TE codes. First of all, the name of the drug product will be given, erythromycin. Then its dosage form will be given, a tablet, and it is given by which route, oral. Then you will have the TE codes. Then you will have how many companies those drug molecules are manufacturing. Then you will have a dose. Then this A2136 is nothing but one kind of a number which has been used by US FDA for their official records. And besides that number, you will have some date. This date talks about when that drug product was registered under the drug product list. So this is the basic information which has been given down into the Orange Book. Now, if I see, there are 3 companies which are manufacturing this erythromycin. One is Cadila Pharma, second one is Teva Pharma, and third one is Torrent Pharma. So all these are having one code AB. Now this signifies, or what is the understanding which the pharmacist has to understand, that if, for example, a patient has been in the prescription, a Cadila Pharma 250 mg erythromycin is being given. Now if basically that drug molecule is not been available, then we can, or the pharmacist can substitute a Teva pharmaceutical erythromycin tablet, which is a 250 mg, or he can substitute the 250 mg tablet of Torrent. So this is basically, it considered that all these drug products are bioequivalent. So this is basically use of this Orange Book. Now I am also having one more company that is called as Amneal Pharma. Now the code is BX. Now as I've already told you that this is not therapeutically equivalent, means generic version is not available. So you can't replace this Amneal Pharmas with the Cadila Pharma, because the coding is different and you can't replace. So this is basically the information which is being given down into the Orange Book. Now during the TE codes, which I've told you that the code is basically a 2-letter code. So sometimes a letter is also attached with that code. For example, AB1 and AB2. Now, what is this 1 and 2 talks about? When there are different drug molecules, more than 2 or more than 3 reference listed drug, means there are multiple companies which have a patented product of the same molecule. For example, I'm having metformin hydrochloride. These are the companies, for example, Sun Pharma to Unichem, then metformin hydrochloride of Ajanta. So if there are multiple branded products, so at that time, the letters are being given with the 2-letter code. So that is AB1 or AB2. Now, This is the example which I have taken. So metformin hydrochloride, which has been used to type 2 diabetes, and it is an extended release product. Now, this basically information shows that if a pharmacist want to go for a substitution, so if a Sun Pharma generic version is given for 500 mg, he has to submit from this group only. He can't substitute from AB1 to AB2. Why? Because whenever these pharmaceuticals have compared the bioequivalent studies, they have taken the standard from this group, not from this group. So this is basically to show that each product of that class is been basically comparable with the branded product. So this is codes which have been given into the Orange Book. I hope so. The things are clear with respect to the different codes which have been given to the each products along with the numbers. Nowadays, basically, this Orange Book is also available on the US FDA website. You have to just type in the Google Orange Book, then this and the first search will help you to reach to the official website of the Orange Book. Now here you can search by different categories, or you can search by different ways. You can search by applicant, you can search by dosage form, you can search by oral, or you can search by route. If you just type that route, all number of search it will display, right? So this is called as an electronic version, means e-copy of the Orange Book. Now I'll just take one example that I have basically put one search that is called as atorvastatin calcium onto this electronic version of the Orange Book. So this will give you similar information which has been there in the Orange Book. It will give market status, whether it is there in the market. It will talk about the active ingredient. It will talk about the name of the company or the proprietary name, Lipitor. Then it will talk about the application number, then the dosage form, then the oral, then the strength, then it will also give you the TE code. Now TE codes, now these products, they are having AB. Now this AB stands about these are therapeutically equivalent. It means we can go for generic substitution for this drug molecule. So this is information can be obtained on the official website, or you can get this information from electronic version of the Orange Book. Now I have again put one I just wanted to show another example. Ticagrelor is one molecule which I have searched into these electronic version of the Orange Book. So Brilinta is a proprietary name. Then it has shown the application number, the dosage form, the route, but here the TE code is not given. So if no TE code is given, then it is being considered that a generic version is not available for that drug molecule. So in this way, basically, we can search whether a generic version is available for a particular product or not. So the Orange Book is updated monthly with the generic version, with generic drug approvals, keeping consumers current on approved drugs in essentials to the issuance of alternate drugs in the event that substitutes have to be prescribed. Now whatever I've told you about the Orange Book, this Orange Book will have only the conventional drug molecules. Now US FDA also published another book. Now that book is called as Purple Book. Now this Purple Book will contain only the biological products. It will not contain the normal products. So this Purple Book is nothing but it is an FDA licensed biological products which has been regulated by a Center of Drug Evaluation and Research. And this Purple Book will give you an idea about 3 drug products. What is biosimilar, what is biological products, and what is interchangeable products. Now, if you talk about biological products, there are different kinds of vaccines comes under biological product, blood and blood components come under biological products, gene therapies, monoclonal antibodies, these recombinant DNA technologies or recombinant proteins. So these huge, large, complex molecules comes under biological products. Now, just wanted to explain you, biological products are nothing but those products which are obtained from biotechnology using living systems such as microorganisms, plant or animal cells to produce it. So purple book will only have biologicals, biosimilars or interchangeable drugs. It will not have a normal version of a tablet capsule. No. And why basically this purple book I'm explaining you, because this purple book will also give an idea about the generic version of the biological product. So FDA has different books. One is the orange book, which talks about the normal drug products, their patented products and the generic version. The purple books talks about the biological product and its generic version. Now, if I want to compare the normal product with the biological products, so this is the graph. Now, the smallest molecule we are talking about, it is aspirin, then this is insulin, this is erythropoietin, and this is monoclonal antibody. So it is large complex molecules. They are obtained from a living systems, maybe from a cell or from a plant stem or from a human cell. And they are basically a lot of complex molecules, very tricky molecules to basically manufacture. And the examples which I've already told you, they are proteins, vaccines, and monoclonal antibodies. Now, in these purple books, you will find three kinds of an product. First product is referred to as reference product. In short form, it is called as RP. Then second product is called as biosimilar product. Now, reference product, if I want to compare with the orange book, this reference product is nothing but I'm talking about the branded drug product. So there are some companies which manufactures or which are involved in research, and they are the first companies to put a biological product into the market. So these products are referred to as reference product. Now, after the patent has expired, then the pharmaceutical companies put a generic version of a biological product into the market. So that biological product is referred to as biosimilar. Got it? So this biosimilar is nothing but a generic version, but it is not named as generic, but because of the products are biological in nature, so it is referred to as a biosimilar product. And then the third one is called as interchangeable products. Now, interchangeable products are such products. For example, if I want to give an example that, okay, one patient is there and one product is being given or one reference product is being given for the treatment, got it, for a period of 6 months. Now, there are some interchangeable drug products which is having the same activity of the reference product. The doctor can there is no need to go to the doctors. The pharmacist can see into this purple book if an interchangeable drug is available. So the pharmacist looking into the this purple book, he can directly substitute these interchangeable product in place of this reference product. Got it? So these three kinds of products are being available in the purple book. So this is called as the interchangeable products. Now, I just wanted to give an example of a disease when biological products are being used. So as you know that RA, rheumatoid arthritis, it is an autoimmune disease. It is an inflammatory disease. Generally, to treat RA, we go for normal NSAIDs, for example, ibuprofen, and this ibuprofen basically will cause the reduction in the pain and the inflammation. Whenever you talk about this RA, there is a swelling of all the joints takes place into the body. If that is not been solved, then we go for DMRDs, that is disease modifying anti-rheumatic drugs. Example is methotrexate. If these drugs are not, you can say, they are not sufficient to recover the disease, then basically we go for biological drug products. Now, this is just a comparison why basically FDA has published the purple book. Now, this is the innovator company where now these all are examples of these are all the examples of monoclonal antibodies, and these are being used to treat the RA. Now, if you talk about a branded drug company, and if you talk about a cost of the treatment for 6 months, if the patient is taking a branded drug molecule, the cost in India for a biological product is 4,80,000. The cost is very huge in terms of biological product. Now, if a patient is being substituted with the generic version, In case of biologicals, it is called as biosimilar. The same cost is been reduced to half. It is 2,40,000. So this purple book will give you an idea about the cost also, and whether a biosimilar is available for the innovators company, so that a pharmacist can basically replace that drug molecule with the innovators company. So this is the PDF format which has been available in the US FDA website, where from that you can download and you can get an information. Now, also similarly to that of an orange book, you can also access these purple book electronically. This is basically the home page of Purple Books, database of licensed biological medicines, where you can just add the letters or any kind of a biological product. It will show the search engine. Now, in this, I've just given you an example that I have added one product that is called as Humira. Humira is nothing but one kind of an monoclonal antibody product which is being used to treat the RA, that is called as rheumatoid arthritis. So it will show different drug molecules. So this is the proprietary medicine, or this is basically the reference product, and below that it will talk about biosimilars. So for this drug product, there are 3 biosimilars products available. So pharmacist can substitute any of the product for this reference drug product. Now, matching cards are there. Matching card means, now in this example you can see that the purple color is there for all the labels. So it indicates that the reference drug product is exactly matching with the generic version, or it is exactly matching with the biosimilar. So this, by looking at the color itself, you will able to know that the biosimilar is available for a biological product. So matching cards color indicates a biological product is similar to the reference drug product. Now, if you just click that label, then again you will get an information. It will talk about the drug name, it will talk about the active ingredients, as I have to, because last 3 MAB, that is called monoclonal antibodies preparation. Then it will talk about the strength, it will talk about the dosage form, it will talk about the prescription. Then this is the biosimilar. The Hulo is one kind of an brand name, or which is the biosimilar of a monoclonal antibody of the Humira. So it will have all the details, right from the dosage form to route of administration to the strength to the product presentation to the license number and the proprietary name. So this was all about the orange book and the purple book. Now US FDA also published one book. That book is called as green book. Now what is this green book? Whatever the veterinary products which are been available, although veterinary products are being published in that green book. So this green book talks about all the list of drug products that have approved by US FDA and which has been used for a veterinary medicine. Now, these veterinary medicine will also have a generic copy. So all the information of the generic version of all the veterinary products you will find in the green book. As I've told you, green it is nothing related to any kind of in 1994, whenever the first copy of this green book was been published, the hardcover was green in nature. So these different colors are being given just to identify which book is which. So orange, it talks about the normal drug product, purple book, it talks about the biological drug products, and green book, it talks about the approved drug products for veterinary use, and it will give you an idea about the generic version of a branded for the branded molecule. Now these are some kind of contents which you will find in the green book. There are different sections from 1 to 8. First, we'll talk about the first section gives an idea about the new drug, animal, if any kind of a new formulation which has been there in the market. So that market, so whatever the product which has been there in the market, that will come into this green book. Then it will talk about the active ingredients. It will give an information about the patented product molecule. It will give an idea about the exclusivity period. Exclusivity period means it will give about the information when the patent is going to expire, or what exclusivity periods are being given to that certain companies with respect to that molecule. Then if any kind of an if the drug molecule is being given to the drug is given to the animal, and if there are certain kind of an adverse reaction, so products subject to notice of hearing means it will have the list. that such products are in the court of the US FDA. It is hearing has been going on because of the safety concerns in the animals also. So then there are the list of voluntary withdrawals, also the petition actions which is taken by the company and the US FDA. So all these information you can get into this green book. So these are the books which are official in most of the foreign countries. Here I have just taken one example of US FDA. They have orange book, green book, and the purple book. Now, one of the important thing is that when one should not go for substitution, one of the most important criteria. So these are 3 things or 4 criteria one should not go for substitution, or generic version should not be substituted for the branded drug molecule. And what those criteria are: clinically significant intolerance to the inactive ingredients. So it means that कि अगर जेनरिक वर्जन में कुछ इनएक्टिव इंग्रेडिएंट्स है और वो इनएक्टिव इंग्रेडिएंट्स किसी पेशेंट में intolerance है, मतलब they are showing some adverse reactions. Correct. So adverse reactions may be skin rashes, maybe paining in the heart, maybe redness throughout the body. So if such cases happens, then the generic version should not be substituted, because this effect is not because of the drug molecule. The effect is because of the inactive ingredients, and these inactive ingredients can be, the effect is shown basically in few people, but in some people it may not show the effect, but in some people, if the people are prone to that inactive ingredient, the side effects can be seen. So if such is the case, please do not substitute a generic version for that drug molecule. Correct. Now, the consumers who are likely to become confused, or some people are vision impaired, जिसको communication हम वो कर नहीं सकते. So in that case, generally, in order to avoid the confusion, generic should not be substituted. If the drugs are having narrow therapeutic index, means the drugs are very potent. Got it? So in that case, generally we should not substitute these generic version. And whenever the consumer has a negative attitude to the submission. So if the consumer does not wants, because many a times whenever a patient goes to the medical shop and he is asking the same molecule which has been written down onto the prescription, but many a times pharmacist is forcing a generic version for his profit. So in that case, if a negative attitude is there in terms of for the patient, so please do not substitute these generic version for those molecules. Now, this is all what the aspects which are been there. Now, it is not so that India is lacking behind. India has also basically but India has not India is not having these any kind of an books available in form of an segregated form, but all the generic version, it is available in the regulatory body of the Indian regulatory body, CDSCO. If you want to search, they will have all the list of generic version. They will have all the list of biological products. So the generic version in India by Sri Narendra Modi ji sir, Honorable Prime Minister of India. So what was their vision? To bring down the healthcare budget of every citizen of India through providing quality generic medicine at affordable prices. So for the promotion of the generics in India, Government of India has introduced the Jan Aushadhi scheme. Now, nowadays, apart from these Government of India has also, or these regulatory body of India, Jan Aushadhi Sugam, a software or a mobile application has been developed. So you can download, and this application can give you a lot of information. What are the generic versions available? What are the price? What are the prices of the generic version with the branded version? So comparison with the generic as well as branded medicine in form of MRP and overall savings are available into this in this mobile app. So this Jan Aushadhi Sugam mobile apps, it will give you search all Jan Aushadhi generic medicines and stores at the fingertip. Also, it will give an idea about the product comparison of generic versus MRP and overall savings. Now, this is the situation in India and US market. In US market, patented drugs are being sold, and the off patents generally are sold in the generic name, which helps making pure generics cheaper. But mostly in India are generics as a brand name generics and a generic name generics. So this is the difference in India. So we have a branded name generics and normal generics. Apart from these, basically, if you are comparing the India and US, in developed countries like US, community pharmacies plays an important role in dispensing the medicines. Hence, their cost awareness becomes crucial because of the well-defined structure and because of all these well-defined books which have been available, and community pharmacies helps the patients that which products you can choose in terms of reduction in the total cost of the product. But it is not so. India is also taking several steps into this regard, and they are basically providing each and every information on the website, provided it is not in a compiled way as in case of Orange Book, Purple Group, or in case of a Green Book for the veterinary drug products. Now, this is the final conclusion from Indian perspective. Now, we have the guidelines. Got it? We so not only enhancing the guidelines, quality drugs in the market, not only providing guidelines will not enhance the quality in the market and subscribing. On the other hand, ensuring the quality with strict regulatory mandate and providing the updated information regarding the generics, for example, as given by US FDA in the Orange Book, will eventually enhance the prescription of generic drugs. Got it? So these generic drugs, if a compilation of copy is been available, it will serve as a book for the information to the state health agencies, prescribers, doctors, and also it will give an help to the pharmacies. So this is where I would like to conclude my presentation, where it if any questions are there, I'm happy if I can answer those.
Thank you so much, sir, for such an informative session.
Thank you.
There is so much interactive session and people are very active. So many questions are there, but because of during the we are running out of the time, I would like to interpret a few of them. First is the substitution of generic drug is good practice. Why?
Substitution of generic is a good practice because I have already told you there is no difference between a generic and a branded drug product. Whenever a company puts a generic version,
hello, sir. Okay.
Sir, unmute your microphone.
Sorry for the interruption because my battery backup has gone. The question was, is it safe that we should go for substitution for a generic version for a branded drug product? Now, the answer is that, see, the regulatory bodies have approved that generic version is equivalent to the branded drug product. And the companies are doing bioequivalence studies in human beings, and they are claiming that it is safe. So, if I want to give an example of a patient who is taking anti-diabetic tablet, if a branded drug product is being given to him or her, and this anti-diabetic tablet he has to take for his lifelong. So if you see the cost will be huge. So in order to make the cost of the tablet less burden for him, if a generic is available for that, we can go for the substitution in terms of cost of the reduction, because efficacy, safety is same as that of the branded drug product. I hope so. I'm able to clarify the answer for the asked question.
Thank you so much, sir. Sir, another question is there any right pharmacist have that he can replace brand medicine to generic medicine to give patient from doctor's prescription?
Actually, see, if I'm talking about USA, they have the clear-cut rules that they can refer orange books and they can go for substitution from generic to branded, depending upon the information which has been available in orange, purple book, or veterinary product. In case of green, in case of the green book. But in India, as such, the stigma is that actually rules are there that we can substitute. But here, the Indian in case of Indian, the scenario is different. If we substitute basically the doctors feel does not good that a pharmacist has replaced and he has got more knowledge about the medicine and he has substitute that drug. So that complications are arising in Indian scenario. But basically, if a generic version is there and if you want to confirm that, you can go on the Indian regulatory website where a generic versions are available and you can substitute for a branded drug molecule, provided the inactive ingredient should not have any adverse effect into the body. So that has to be checked before giving the generic version. I hope so. The things are clear.
Thank you so much, sir, for such an information session. And participants really enjoyed it. From their question, we can interpret it. Okay, now moving towards our validatory function. Technology won't replace teacher, but teachers who use technology will probably replace teacher who do not. So I would like to introduce guest of honor of today's validatory function, Mr. Jasubhai Chaudhary, Registrar, Gujarat State Pharmacy Council, Ahmedabad. I request Mr. Jasubhai Chaudhary, sir, to say a few words. Sir.
All right, thank you very much, ma'am. Actually, I'm traveling on my way to somewhere, so I'll take only 5-10 minutes and will conclude my speech. So, well, I could see almost 200 participants have joined these 2 days refresher course. That means almost 200 participants have taken benefit of the lectures, very nicely selected topics by the Indu Kaka Koala College of Pharmacy. I was listening to the last lecture of Sri Ashok Mahajanji. He was, he was providing a very informative speech also. So I think entire refresher course, which delivered almost 7 lectures, must have been very beneficial to the entire pharmacy friends who have joined. And I take an opportunity today to convey my sincere thanks to the principal and the entire team of Indu Kaka Koala College of Pharmacy, who have been working very hard, who have been continuously supporting the State Pharmacy Council in organizing such kind of refresher courses. The refresher courses are a platform who continuously help us to upgrade the knowledge of our refresher, knowledge of our pharmacist friends. And ultimately, they are a very handy tool to upgrade the knowledge and ultimately serves a very useful platform for upgradation of the entire profession also. But before I say anything about State Pharmacy Council, I take an opportunity to convey the things about the renewal process, because most of the pharmacist friends who have joined today, they might be having their renewals due next year. મોટાભાગના pharmacist મિત્રો renewal ના કારણે refresher course માં ભાગ લેતા હોય છે, પરંતુ once they participate in the refresher course, they realize કે માત્ર renewal કરવા માટે આ refresher course કરવો પડે એના કરતા વધારે ઉપયોગી થતો હોય છે. 7 lectures જે સિલેક્ટ થતા હોય છે કોલેજ દ્વારા, અને જે speakers that they elect or select, I would say, for this refresher course, they are very experienced ones. અને એના કારણે ultimately 7 lectures ભલે એક formality લાગે કે રિફ્રેશર કોર્સ આપણે રિન્યુઅલ માટે કરવું પડે છે. પણ જે મિત્રો રિફ્રેશર કોર્સ પાર્ટિસિપેટ કરે છે અને એના પછીના જે રિવ્યુઝ આવે છે એમના ચેકબોક્સમાં, ત્યારે વી કેન રિયલાઇઝ કે ખરેખર રિફ્રેશર કોર્સના આજે 7 લેક્ચર્સ છે એના માધ્યમથી દે હેવ ગેઇન્ડ નોલેજ. ઇવન જ્યારે હું પણ સાંભળતો હોઉં છું ત્યારે એવું લાગે છે કે વર્ષો પહેલા જે આપણે ક્યાંક આપણો અભ્યાસ છોડી દેતા હોઈએ છીએ, એટલે મતલબ કમ્પ્લીટ કર્યો હોય છે, દેન આફ્ટર આપણે આપણી જાતને આપણા પ્રોફેશન રિલેટેડ અપડેટ નથી કરતા, અને એટલા માટે ક્યાંક ને ક્યાંક આપણે એ બધાથી પાછળ છૂટતા હોઈએ છીએ. અને એટલે જ રિફ્રેશર કોર્સ આપણને એક તક આપે છે, એક ટર્મિનોલોજી, એટલીસ્ટ ધ ટ્રાન્સફોર્મેશન્સ ધેટ આર હેપનિંગ ઇન ધ પ્રોફેશન, અથવા તો જે અત્યારની અપડેટ્સ છે એ મેળવવાનું એક લેટેસ્ટ ચાન્સ હોય તો એ રિફ્રેશર કોર્સ છે. અને સ્ટેટ કાઉન્સિલ્સની વાત કરીએ તો આપણી સ્ટેટ કાઉન્સિલે આ જે પ્રવૃત્તિ છે અથવા તો એક્ટિવિટી છે એને એકદમ સિરિયસલી લીધેલી છે. અને એવરી યર વી કન્ડક્ટ, હવે તો એવું છે કે અત્યારે એવરી વીક ચાલે છે, અને જાન-ફેબ-માર્ચ જ્યારે આપણો રિફ્રેશર કોર્સ રિન્યુઅલનો પીરિયડ આવશે ત્યારે એવરી વીકેન્ડ આપણે 2 રિફ્રેશર કોર્સ ચલાવી છે. અને ઇન ધેટ વે આપણી સ્ટેટ કાઉન્સિલે ઇન્ડિયાની બધી જ સ્ટેટ કાઉન્સિલ્સમાં રિફ્રેશર કોર્સ કરવામાં નંબર 1 પર છે. અને એનું ધ રીઝન ઇઝ બીકોઝ the support that we are getting from the institutions like Induka Kaikwala College of Pharmacy. તો આ બધાના સપોર્ટથી આપણે, લાઈક ઇન મેની અધર એક્ટિવિટીઝ, આપણી સ્ટેટ કાઉન્સિલ ઇઝ અ મોડેલ કાઉન્સિલ ફોર અધર સ્ટેટ કાઉન્સિલ્સ ઇન ઇન્ડિયા. પરંતુ મારો આજનો હેતુ બધાની વચ્ચે આવવાનો એ છે. આઈ એમ ટ્રાવેલિંગ ઓલ્સો, બટ મને લાગ્યું કે મારે 10 મિનિટ આપની જોડે વાત કરવી જોઈએ, કારણ કે આપની બધાની રિન્યુઅલ પ્રોસેસ ડ્યુ છે, જે લોકોનું 31 ડિસેમ્બર 2023માં પૂર્ણ થાય છે વેલિડિટી, એ બધાના રિન્યુઅલ્સ કરવાના થશે 1 જાન્યુઆરીથી 2024 થી 31 માર્ચ 2024 ની વચ્ચે. અને આ સમયગાળામાં જે રિન્યુઅલ્સ થવાના છે મિત્રો, એ પ્રોસેસ આગળ જે તમે કરેલા છે એનાથી એન્ટાયરલી ચેન્જ છે. કારણ કે આપણી કાઉન્સિલ એ હવે ઇટ્સ અ પેપરલેસ એન્ડ કેશલેસ કાઉન્સિલ્સ. પહેલી કાઉન્સિલ ઇન ઇન્ડિયા એવી બની છે કે વિચ ઇઝ પેપરલેસ એન્ડ કેશલેસ. વી આર નોટ એક્સેપ્ટિંગ એની પેપર્સ. તમારે ક્યાંય પણ કોઈ પણ ઝેરોક્સનું, એફિડેવિટ્સ કશું જ આપવાનું નથી ઇન પેપર. વી આર એક્સેપ્ટિંગ એવરી પેપર્સ ઓનલાઈન, એ ઘરે બેઠા. એન્ડ કેશલેસ ઇન ધ સેન્સ વી આર નોટ એક્સેપ્ટિંગ એની ડ્રાફ્ટ્સ, એની ફીઝ ઓવર ધેર ઇન ધ કાઉન્સિલ્સ. તમારી ફી પણ ઓનલાઈન, યુ આર પેઇંગ ફ્રોમ યોર હોમ. તો ધીસ ઇઝ ફર્સ્ટ મોડલ કાઉન્સિલ ઇન ઇન્ડિયા વિચ ઇઝ પેપરલેસ એન્ડ કેશલેસ. અને એટલા માટે આઈ વોન્ટ ટુ એક્સપ્લેન યુ કે આ વખતનું રિન્યુઅલ તમારું કઈ રીતે જવાનું છે. હું ટ્રાય કરીશ લાસ્ટ ટાઈમનું તમારું રિન્યુઅલ અને આ વખતનું રિન્યુઅલ એટલા માટે કમ્પેર કરવા પ્રયત્ન કરું છું કે જેથી કરીને લાસ્ટ ટાઈમ કરતા આ વખતમાં ચેન્જ શું આવ્યો છે. મિત્રો, જ્યારે લાસ્ટ ટાઈમ તમે રિન્યુઅલ કર્યું છે, તમે બધા કાં તો અમદાવાદ આવ્યા છે, ઓલ ઓવર ગુજરાતમાંથી બધા લોકોને અમદાવાદ ટ્રાવેલ કરવું પડતું હતું. ત્યાં આવીને રિન્યુઅલ્સની ક્યુમાં ઉભા રહેવાનું, ડોક્યુમેન્ટ્સ આપવાના, ફોર્મ ફિલઅપ કરવાનું, ફોટોગ્રાફ એટેચ કરવાના, ક્યાંય ડુપ્લિકેટની જરૂર હોય તો એફિડેવિટ્સ આપવાના, અને અલ્ટિમેટલી ફી ભર્યા પછી એની પહોંચ મેળવતા સાંજના 4-5 વાગી જાય ત્યારે તમને એવું લાગે કે મારું રિન્યુઅલની પ્રોસેસ પૂરી થઈ છે. એ સિવાય કેટલાક મિત્રો જે ટ્રાવેલ નથી કરી શકતા અમદાવાદ, એ બધા લોકો પોસ્ટથી એમના ડોક્યુમેન્ટ્સ મોકલતા હતા. તો પોસ્ટમાં પણ કેટલાકના ડોક્યુમેન્ટ્સ મળે ન મળે. અચ્છા, પોસ્ટમાં એમનું રિન્યુઅલ કર્યા પછીની રિસિપ્ટ તમારા ઘરે અમે મોકલતા હતા. તો તમારા એડ્રેસ અપડેટેડ ના હોય અમારા ત્યાં, તો એના લીધે તમારા રિન્યુઅલ્સની રિસિપ્ટ પાછી આવતી હતી. તો 2 વર્ષ 3 વર્ષ સુધી બધા ફાર્માસિસ્ટ મિત્રોના ફોન આવે કે સર અમને રિન્યુઅલ રિસિપ્ટ મળી નથી. તો એ બધું શોધવાનું, અને અલ્ટિમેટલી ધીસ વોઝ વેરી કન્ફ્યુઝિવ પ્રોસેસ. પરંતુ મિત્રો, આ વખતે મતલબ 2022 થી લાસ્ટ યર અને આ વર્ષે 22 અને 23 2 વર્ષ આપણે ઓનલાઈન રિન્યુઅલ્સ કર્યા છે. અને આ વખતે અગેન 24માં પણ એ થોડા રિફોર્મ્સ સાથે વી આર કમિંગ with online renewal process that would make a very easiest process for you. મિત્રો, આ વખત જે રિન્યુઅલ થઈ રહ્યું છે એટલા માટે સમજાવું છું કે તમારે ભલે જાન્યુઆરી, ફેબ્રુઆરી, માર્ચમાં કરવાનું છે, પરંતુ લોકો ઇન એડવાન્સ ડ્રાફ્ટ્સ કઢાવી લેતા હોય છે ડિસેમ્બરમાં, અથવા તો પછી ત્યાં આવીને ડોક્યુમેન્ટ્સ મોકલાવતા હોય છે. પરંતુ આ વખતે મિત્રો, જેમ તમે રિફ્રેશર કોર્સનો આનંદ લઈ રહ્યા છો ઘરે બેઠા, જેમ ઘરે બેઠા લિંક ભરી છે, ઘરે બેઠા ઝૂમની લિંક મેળવી છે, અને ઘરે બેઠા જ લેક્ચર ભર્યા છે, એવી જ વ્યવસ્થા રિન્યુઅલમાં છે. રિન્યુઅલમાં પણ આપણી ગુજરાત સ્ટેટ ફાર્મસી કાઉન્સિલની વેબસાઇટમાં એકાઉન્ટ બનાવી લેવાનું છે મિત્રો. જેમ તમે ગૂગલ પે, ફોન પે કે બાકી બધા બેન્કિંગ, નેટ બેન્કિંગનું એક વખતે તમારું એકાઉન્ટ ક્રિએટ કરી લેતા હોય છે, તમારું આઈડી પાસવર્ડ બનાવી લેતા હોય, અને પછી તમારે પૈસા ટ્રાન્સફર કરવા છે કે વોટ એવર સ્મોલ થિંગ્સ આર ધેર, એ બધું એકદમ ઇઝી છે. એવી જ રીતે આપણી વેબસાઇટમાં આપણું એકાઉન્ટ એક વખત બનાવી લ્યો છો, એના પછી રિન્યુઅલ કરવું બહુ સરળ છે મિત્રો. ઘરે બેઠા બંને ડોક્યુમેન્ટ્સ અપલોડ કરવાના છે. ઓનલી 2 documents we are asking. One is your refresher course certificate અને એક આધાર કાર્ડ. આ બે ડોક્યુમેન્ટ પીડીએફ માં અપલોડ કરવાના છે. નેક્સ્ટ સ્ટેજમાં 550 રૂપિયા ઓનલાઈન ઘરે બેઠા ડેબિટ કાર્ડ, ક્રેડિટ કાર્ડ કે એની યુપીઆઈ આઈડી કે તમારા એની યુપીઆઈ વોલેટ્સ હોય તમારે, એમાંથી તમે ઘરે બેઠા પે કરી શકો છો. વિધિન 1 or 2 minutes આ તમારી પ્રોસેસ પૂરી થઈ જાય છે. અને તમે તો એડવાન્સ ચાલી રહ્યા છો, તમારું રિફ્રેશર કોર્સનું સર્ટિફિકેટ અઠવાડિયામાં તમને મળી જવાનું છે. તો 1 જાન્યુઆરી જ રાત્રે 12 વાગે બેસી જાઓ, 12 ને 1 તમારું સબમિટ કરી દો તમે ઘરે બેઠા. ઇવન નાઇટ ટાઈમ પણ તમે કામ કરી શકો છો. એવરીથિંગ ઇઝ ઓનલાઈન છે. મતલબ તમારે ઓફિસેથી ઘરે આવીને ઘરના અનુકૂળતાના સમયે તમારા ડોક્યુમેન્ટ્સ અપલોડ કરો, પેમેન્ટ કરો, અને એ પેમેન્ટ થયા પછી મિત્રો તરત જ તમને એક રિસિપ્ટ મળવાની છે. પૈસા મળ્યાની રિસિપ્ટ તરત જનરેટ થવાની છે. પરંતુ મિત્રો લાસ્ટ ટાઈમ કરતા ફરક એટલો છે કે લાસ્ટ ટાઈમ જે તમે અહીંયા આવીને પૈસા ભરતા હતા, એ પૈસા ભર્યાની પહોંચની અંદર જ એવું લખીને આવતું હતું કે તમારું 5 વર્ષ સુધીનું રિન્યુઅલ થયું છે. આ વખતની રિન્યુઅલ પ્રોસેસ આપણે 2 ભાગમાં ડિવાઈડ કરી છે મિત્રો. તમારું જે રિન્યુઅલ ના તમે પેમેન્ટ કરવાના છો, 550 રૂપીઝ ધેટ યુ આર ગોઈંગ ટુ પે, એની રિસિપ્ટ ઇન્સ્ટન્ટ જનરેટ રાત્રે 12 વાગે પણ તમે પે કરો છો તો ઇન્સ્ટન્ટ જનરેટ થઈને તમને રિસિપ્ટ મળવાની છે એમાં. પરંતુ એ રિન્યુઅલની પહોંચ નથી મિત્રો. ઇટ્સ નોટ ધ રિન્યુઅલ પ્રૂફ ઓફ રિન્યુઅલ. એ 2 ભાગમાં ડિવાઈડ કર્યું છે. પૈસા ભર્યાની પહોંચ અને રિન્યુઅલનું સર્ટિફિકેટ આ વખતે અલગ પાડ્યું છે 2 વર્ષથી. તો તમે જે પૈસા ભરશો એની પહોંચ તરત મળશે. તમારા પૈસા સ્ટેટ ફાર્મસી કાઉન્સિલ સુધી પહોંચ્યા છે. એક પહોંચ તમને પેયુ મનીની મળશે, વચ્ચે જે ગેટવે પૈસા કલેક્ટ કરે છે, અને બીજી પહોંચ તરત જ સ્ટેટ ફાર્મસી કાઉન્સિલની મળે છે તમને કે ભાઈ 550 રૂપીઝ હેવ બીન રિસીવ્ડ બાય સ્ટેટ ફાર્મસી કાઉન્સિલ. પરંતુ તમારું એ રિન્યુઅલ નથી. રિન્યુઅલ ક્યારે થાય છે? બીજા દિવસે અથવા તો વર્કિંગ દિવસે. જ્યારે તમે દિવસે કરો છો અને વર્કિંગ દિવસ હશે, ઓફિસ અવર્સ હશે, તો અમારી ઓફિસ એ ડોક્યુમેન્ટ ચેક કરશે. બંને ડોક્યુમેન્ટ પ્રોપર છે. આધાર કાર્ડ એન્ડ રિફ્રેશર કોર્સ સર્ટિફિકેટ એઝ ડિઝાયર્ડ અપલોડ કરેલું છે અને ફીસ ભરેલી છે. તો વિધિન 1 minute વિલ અપ્રૂવ યોર રિન્યુઅલ એપ્લિકેશન. અને રિન્યુઅલ એપ્લિકેશન એપ્રૂવ થશે તરત જ તમને એક સર્ટિફિકેટ ઓનલાઈન મળવાનું છે. તરત જ એક મેલ આવવાનો છે, જે મેલમાં તમારું રિન્યુઅલ સર્ટિફિકેટ આવી જવાનું છે મિત્રો. અને જેમાં લખેલું હશે કે મિસ્ટર XYZ G number this is and his renewal has been his registration has been renewed up to 31st December 2029. આવું એક સર્ટિફિકેટ ઓનલાઈન આવવાનું છે. તો હવે કંઈ પણ તમારે ત્યાં મેળવવા માટે અમદાવાદ આવવાની જરૂર નથી. નાઈધર કંઈ પણ પોસ્ટથી આવવાનું છે. પોસ્ટથી કઈ રાહ જોવાની નથી. આ તમારું રિન્યુઅલનું સર્ટિફિકેટ ઓનલાઈન જ આવવાનું છે. ઓનલાઈન ડાઉનલોડ કરવાનું છે. અચ્છા, એ ખોવાઈ જાય છે. પહેલા એવું થતું કે એકવાર ખોવાઈ ગઈ રિસિપ્ટ રિન્યુઅલની તો ફરી અમદાવાદ આવવાનું, ડુપ્લિકેટ માટે અરજી કરવાની, એના માટે એફિડેવિટ કરવાનું. આ બધી ઝંઝટમાંથી મુક્તિ છે મિત્રો. ખોવાઈ જાય છે તો ફરીથી એ જ તમારો આ બધો જ રેકોર્ડ, આજ પછી તમે જે કંઈ પણ પૈસા ભરવાના છો, જે કંઈ પણ તમારા રિન્યુઅલના કે રીએન્ટ્રીના કે ડિગ્રી એડિશનના બધા સર્ટિફિકેટ જનરેટ થવાના છે. એ બધા જ સર્ટિફિકેટ તમારી વેબસાઇટમાં તમે જે એકાઉન્ટ બનાવેલું છે એનો આઈડી પાસવર્ડ તમારે યાદ રાખવાનો છે. એમાં જમા રહેવાના છે. એકવાર ખોવાઈ જાય છે અથવા તમે ઘરે પડ્યું છે, માર્કેટમાં છો, ડ્રગ ઓફિસ જવું છે, ત્યાં તમારે રિન્યુઅલનો પુરાવો આપવો છે. વન્સ અગેન યુ કેન ડાઉનલોડ ફ્રોમ યોર વેબસાઇટ અને ત્યાં પ્રિન્ટ લઈ શકો છો. તો એઝ મેની ટાઈમ્સ એઝ યુ વિશ ટુ ગેટ ધ પ્રિન્ટ ઓફ યોર રિન્યુઅલ સર્ટિફિકેટ, you can print it. You don't need to apply for duplicate receipt or duplicate certificate. તો મિત્રો આ પ્રકારની સુવિધા આ વખતે થઈ છે. તમારા ભૂતકાળના અનુભવો, તમે ફ્રેશ રજીસ્ટ્રેશન પણ કર્યું છે ત્યારે આપણે યાદ છે અમદાવાદ જવાનું, lot many forms had to be filled અને એફિડેવિટ્સ ના બધી પ્રોસેસ હવે બંધ છે મિત્રો. એમના માટે પણ સરળ પ્રક્રિયા છે. અત્યારે જે નવા વિદ્યાર્થીઓ ડિપ્લોમા, બેચલર ફાર્મસી કે ફાર્મડી કરીને પાસ આઉટ થાય છે, એ લોકો માટે પણ આવી સુવિધા છે. એમને એક વખત કાઉન્સિલ આવવું પડે છે વેરિફિકેશન ઓફ ઓરિજિનલ ડોક્યુમેન્ટ્સ માટે અને એમના ફોટોગ્રાફ માટે. પરંતુ એ પહેલા બધા જ ડોક્યુમેન્ટ એ લોકો ઘરે બેસીને ઓનલાઈન અપલોડ કરે છે. ડિટેલ્સ બધી ઓનલાઈન ભરાય છે. ફીઝ ઓનલાઈન ભરાઈ જાય છે. એપોઇન્ટમેન્ટ એ લોકો સિલેક્ટ કરે છે ને એપોઇન્ટમેન્ટના દિવસે એ લોકો ત્યાં આવે છે અને ત્યાં વિધિન 15 મિનિટ્સ મિત્રો એમનું ઓરિજિનલ ડોક્યુમેન્ટ્સ વેરિફિકેશન એને બધું જ એ પૂર્ણ થઈ જાય છે. તો એના કારણે અને હવે પ્રક્રિયા એવી છે, એટલી સરળ થઈ છે ટેકનોલોજીના કારણે કે નવું રજીસ્ટ્રેશન પણ સમજો કે પ્રોવિઝનલ હોય તો વિધિન અ ડે એમનું રજીસ્ટ્રેશન સેમ ડે થાય છે. અને 7 સુધી તો એમનું પ્રોવિઝનલ સર્ટિફિકેટ એમના મેલમાં ચાલુ જાય છે. જેમ તમને આજે રિન્યુઅલ સર્ટિફિકેટ મળવાનું છે, એવી જ રીતે સેમ ડે એમને પ્રોવિઝનલ સર્ટિફિકેટ મળી જાય છે. ઓરિજિનલ સર્ટિફિકેટ વિધિન અ ડે ડિસ્પેચ થાય છે, અથવા તો મોસ્ટલી બની શકે છે તો સેમ ડે ત્યાંના ત્યાં એમને પ્રિન્ટ કરીને આપી રહ્યા છે મિત્રો. તો ક્યાં ભૂતકાળની અંદર 1-1.5 મહિના સુધી આપણે રજીસ્ટ્રેશન સર્ટિફિકેટની રાહ જોવી પડતી હતી, એ આજે ટેકનોલોજીનો ફાયદાને લીધે our council has implemented as many technology as we can એ કરી રહ્યા છે, હજુ પણ એમાં સુધારો કરી રહ્યા છે. તો અલ્ટિમેટલી એ ફાયદો આપણા ફાર્માસિસ્ટ મિત્રોને થઈ રહ્યો છે. રજીસ્ટ્રેશન, ન્યૂ રજીસ્ટ્રેશન હોય કે રિન્યુઅલ હોય, હવે તો બાકીની બધી પ્રોસેસ હોય, તમારું ડિગ્રી એડિશન છે, ડિપ્લોમા કર્યું છે, અને એના પછી એના આધારે રજીસ્ટર થયા છો અને બેચલર ફાર્મસી એમ.ફાર્મ કે ફાર્મ.ડી કરી લો છો, તો એનું ડિગ્રી એડિશન પણ ઓનલાઈન થઈ રહ્યું છે. નેમ ચેન્જ છે, આધાર, તમારું એડ્રેસ ચેન્જ. મોસ્ટ ઇમ્પોર્ટન્ટ થિંગ અત્યારના ટાઈમ પીરિયડમાં એ છે કે મિત્રો આપણી સ્ટેટ ફાર્મસી કાઉન્સિલની ઇલેક્શન પણ જાહેર થયું છે. તો તમારા જે બેલેટ પેપર્સ છે એ તમારા ઘરે તમારા એડ્રેસ પર આવવાના છે. તો એડ્રેસ ચેન્જ પહેલા કરવા માટે અમદાવાદ આવવું પડતું, અથવા તો એપ્લિકેશન મોકલવી પડતી હતી. પરંતુ હવે મિત્રો તમને બધાને એ સલાહ આપું છું કે આપણી વેબસાઇટ ગુજરાત સ્ટેટ ફાર્મસી કાઉન્સિલ.org એમાં જઈએ, આપણું એકાઉન્ટ ક્રિએટ કરી લઈએ. એમાં 2 પ્રકારના લોકો એકાઉન્ટ કરી શકે છે. એક એકાઉન્ટ બનાવવા વાળા છે જેનો ઓલરેડી રજીસ્ટર્ડ ફાર્માસિસ્ટ છે, મતલબ તમે બધા લોકો, અને એક જેનું નવું રજીસ્ટ્રેશન બાકી છે. તે 2 ઓપ્શનથી આપણું એકાઉન્ટ બને છે. ત્યાં ભૂલ થાય છે મિત્રો. તો ધ્યાન રાખીએ કે આપણું એકાઉન્ટ બનાવતી વખતે એ ઓપ્શન ક્રિએટ કરીએ કે આપણે ઓલરેડી રજીસ્ટર્ડ છે. એ એકાઉન્ટમાં, એ ઓપ્શનમાં જે આપણું એકાઉન્ટ બનાવી લેશો તો પછી ઘરે બધી પ્રક્રિયા થાય છે, જેમાંનું એડ્રેસ ચેન્જ પણ તમે સરળતાથી ઘરે બેઠા સબમિટ કરી શકશો. તમારું એડ્રેસ બદલાયું છે, મેરેજ પછી બધી બહેનોના લગભગ એમના એડ્રેસ બદલાતા હોય છે, તો નવું એડ્રેસનો પુરાવો અપલોડ કરવાનો છે અને તમે એપ્લાય કરશો તો તરત જ વિધિન અ ડે તમારું એડ્રેસ ચેન્જ આપણી સિસ્ટમમાં ઇફેક્ટિવ થઈ જતું હોય છે. અને આગામી સમયમાં ઇલેક્શનના બેલેટ પેપર પણ એ નવા એડ્રેસ પર અમે મોકલી શકીશું. તો મિત્રો, આ એડવાઇઝ હું તમને બધાને સજેસ્ટ કરું છું કે આજે જ, ભલે તમારું રિન્યુઅલ આવતા જાન-ફેબ-માર્ચમાં હોય, પરંતુ આજે જ એકાઉન્ટ બનાવી લઈએ આપણી સ્ટેટ ફાર્મસી કાઉન્સિલની વેબસાઇટમાં, પોતાનું એડ્રેસ અપડેટ કરી લઈએ, એવરીથિંગ કરી લઈએ, રેડી હશે. અને રિન્યુઅલ પણ 1 જાન્યુઆરીએ તમારું સર્ટિફિકેટ વિધિન અ વીક ઉસ્માન ગની સાહેબને એમની ટીમ એ આપવાની છે. તો એ રેડી હશે. તમે 1 જાન્યુઆરી એપ્લાય કરી લેશો તો આઈ હોપ કે 2 જાન્યુઆરી તો તમારું રિન્યુઅલ થઈ અને સર્ટિફિકેટ તમારા લોગિનમાં વેબસાઇટમાં આવેલું હશે મિત્રો. તો ઇટ્સ બીન વેરી સિમ્પલ. આઈ વુડ હેવ ટ્રાઈડ ટુ ગીવ યુ અ ડેમો ઓલ્સો, પરંતુ એઝ આઈ એમ ટ્રાવેલિંગ, મારી જોડે જે ગેજેટ છે વુડ નોટ મેક મી ટુ પ્રોડ્યુસ અ ડેમો. પરંતુ મિત્રો, મેં એક્સપ્લેન કર્યું છે. ક્યાંય તમને મુશ્કેલી છે તો યુ કેન કોન્ટેક્ટ સ્ટેટ ફાર્મસી કાઉન્સિલ. ક્યાંય નથી સમજાતું તો યુ કેન મેલ અસ. આપણું વેબસાઇટમાં આપણું મેલ એડ્રેસ છે, તમે મેલ કરશો કે મને લોગિનમાં પ્રોબ્લેમ આવે છે, યુ લીવ યોર નંબર ઓવર ધેર. સ્ટેટ ફાર્મસી કાઉન્સિલ સામેથી કોન્ટેક્ટ કરી અને વિલ ટ્રાય ટુ એક્સપ્લેન એન્ટાયર પ્રોસેસ અગેન ઓલ્સો. તો મિત્રો, મને લાગે છે કે સરળ પ્રોસેસ થઈ છે. ઇટ ઇઝ બીકોઝ ઓફ ધ ટેકનોલોજી એન્ડ our entire team which has always supported our state pharmacy council team. તો અને institutions like Induka Kaikowala College have been we have got the support entirely during the year for conducting refresher courses. I once again acknowledge their support and once again convey my sincere thanks to the principal and entire team for supporting the entire fraternity of pharmacists. Once again, I congratulate all the participants who have decided to participate in the refresher course and update their knowledge. I hope કે આવનારા સમયમાં તમે જે કંઈ પણ નોલેજ મેળવો છો એનો સમાજના હિતમાં સદુપયોગ થશે, એ જ ભાવના સાથે would like to conclude my speech. Thank you very much for inviting me in the valedictory function of refresher course. Thank you very much, ma'am.
Thank you so much, sir, for sharing such an informative information about certificate and renewal process and receipt. All participants. Yes, sir.
Yeah. જસુ સર, એક ક્વેશ્ચન હતો જે ઘણા બધા આપણા રજિસ્ટર્ડ ફાર્માસિસ્ટ ફ્રીક્વન્ટલી મને બી કોલ કરતા હોય છે કે જેમને જાન્યુઆરી 2023 થી માર્ચ 2023 માં રિન્યુઅલ રહી ગયું છે.
Yes, exactly, sir.
તો એ લોકો શું કરે એમની માટે થોડું પ્લીઝ.
It's a very important sir, can you please share the screen for me? If you can share the screen like Gujarat State Pharmacy Council. મને એમ કે તમે નથી સાહેબ આજે, એટલે તમે આની જોડે સ્ટેટ ફાર્મ ગૂગલ ખોલી દેશો સર, તો મને લાગે છે ડેમો મારે આપી દેવો જોઈએ. તમે શેર કરો સ્ક્રીન અને ગૂગલમાં આપણી શોધી અને બધાને સમજાવો કે સ્ટેટ ફાર્મસી કાઉન્સિલની વેબસાઇટ છે એ આપણે કઈ રીતે ખોલતા હોય છે. તમને પ્રોસેસ ખ્યાલ છે એટલે હું કહેતો જાઉં અને તમે પેજ જરા સરકાવતા જશો, સ્લાઇડ કરતા જશો તો વુડ બી હેપી. બધા માટે ઈઝી થઈ જશે સર. તો તમને કે સાહેબ તમારી સ્ક્રીન શેર થઈ છે કે મારી થઈ છે? આઈ થિંક યુ હેવ શેર ધ સ્ક્રીન. ઓકે ઓકે ઓકે. રાઈટ સર. ફૂલ પેજ કરજો સાહેબ.
હા હા.
ઓકે મિત્રો, હું બોલતો જાઉં છું. સ્ટેટ ફાર્મસી કાઉન્સિલની વેબસાઇટ છે GSPC, તમે ટાઈપ કરશો તો પણ મળી જશે. ઇટ્સ રાઈટ વેબસાઇટ છે. સી વી કેન સી gujaratpharmacycouncil.org. હા યસ, એના પર ક્લિક કરી લઈએ. તો મિત્રો તમને વેબસાઇટ યાદ નથી રહેતી તો પણ ઇવન ગૂગલમાં, હું એટલા માટે સ્લો અને આ સમજાવવાનું પ્રયત્ન કરું છું કે કેટલાક જૂના ફાર્માસિસ્ટ મિત્રો છે એમને જે લોકો ટેકનો સેવી નથી એમને આ મુશ્કેલી પડે છે, એટલા માટે હું ડેમો પણ આપવાનો પ્રયત્ન કરું. બાકી નવા ફાર્માસિસ્ટ મિત્રો છે એમના માટે ધીસ થિંગ ઇઝ વેરી ઇઝી. અચ્છા, મારો હું પેલો ક્વેશ્ચન લઈ લઉં છું સાહેબ. ઇઝ ઇટ ઓડિબલ?
યસ યસ.
ઓકે, તમે કન્ટિન્યુ કરો. મીનવાઇલ જે પ્રશ્ન સાહેબે પૂછ્યો છે કે લાઈક ડ્યુ હોય છે આપણું, હંમેશા આપણો નિયમ એવો છે મિત્રો રિન્યુઅલનો કે આપણું રજીસ્ટ્રેશન છે ને હંમેશા 31 ડિસેમ્બરની વેલિડિટી ધરાવે છે. જે તે વર્ષના હંમેશા એ 31 ડિસેમ્બરે એક્સપાયર થાય. હવે નિયમ એવો છે કે 31 ડિસેમ્બર 2022માં પૂરું થાય છે તો એનું રિન્યુઅલ હંમેશા એનું જે નેક્સ્ટ યર છે, ફોલોઈંગ યર, એનું 1 જાન્યુઆરીથી 31 માર્ચમાં આપણે એનું રિન્યુઅલ કરવાનું થતું હોય છે. મતલબ કે 31 ડિસેમ્બર 2022 કોઈનું પૂરું થયું છે તો એમને રિન્યુઅલ કરવાનું થાય છે 1 જાન્યુઆરી 2023માં. આપણા બધા લોકોનું આ વખતે પૂરું થાય છે કે 31 ડિસેમ્બર 2023. તો એ બધાનું રિન્યુઅલ એમને કરવાનું થાય. રીલોડ કરી લઈએ સાહેબ, એવું કોઈનું સજેશન છે? નેટ પ્રોબ્લેમ હોઈ શકે. સાઇટ તો બરાબર છે સાહેબ, ગુજરાત ફાર્મસી કાઉન્સિલ. તો સમજો કે 2023માં પૂરું થતું હોય એમનું રિન્યુઅલની એપ્લિકેશન 1 જાન્યુઆરી 2024 થી 31 માર્ચ 2024 થવું જોઈએ, પરંતુ તે મિસ થઈ જાય છે તો શું કરવાનું? 22માં હતું અને 23માં એનું રિન્યુઅલ કરવાનું હતું. સમજો એ 31 માર્ચ 2023 સુધી કરવાનું હતું અને આપણે રિન્યુઅલ કરી શક્યા નથી, તો એવા કેસમાં શું થઈ શકે? તો એકચ્યુઅલી જે રિન્યુઅલ પીરિયડ 31 માર્ચ સુધીનો છે, પછી તમે રિન્યુઅલની અરજી કરી શકતા નથી. અને એવા સમય શું થાય છે, જે તમે સમય દરમિયાન તમારું રિન્યુઅલમાં ફીસ ભરતા નથી તો તમારું નામ ટેમ્પરરીલી સ્ટેટ ફાર્મસી કાઉન્સિલના રજિસ્ટરમાંથી રિમૂવ થાય છે મિત્રો. નામ દૂર થાય છે પણ ઇટ ઇઝ ટેમ્પરરીલી. અને જો તમારે પછી એ ડ્યુ પીરિયડ મિસ કર્યો છે અને ફરીથી તમારું નામ એન્ટર કરવું છે તો તમારે નવું ફ્રેશ રજિસ્ટ્રેશનની અરજી કરવાની જરૂર નથી, પરંતુ તમારે તમારું નામ ફરીથી એન્ટર કરાવવા માટેની, મતલબ રી એન્ટર કરવા માટેની પ્રક્રિયા કરવી પડે અને એ અરજીનું નામ છે રી એન્ટ્રી ઓફ રજિસ્ટ્રેશન. તો આપણે રી એન્ટ્રી વર્ડ સાંભળેલો છે અને હમણાં સાહેબ વેબસાઇટ આપણને ખોલશે તો હું તમને રી એન્ટ્રી પણ બતાવીશ. પણ મતલબ એવો જ સિમ્પલ એ છે કે રિન્યુઅલનો ડ્યુ પીરિયડ છે અને જો તમે મિસ કરી ગયા છો. અચ્છા સાહેબ, એવું બની શકે કે વેબસાઇટ ડાઉન હોઈ શકે સ્ટેટ ફાર્મસી કાઉન્સિલ. યા ઇટ ઇઝ ઓપનિંગ આઈ થિંક. મતલબ મેં કહ્યું એમ, જો તમે ડ્યુ પીરિયડ મિસ કરી જાવ છો, યસ વી ગોટ ઇટ. તો મિત્રો સાહેબ નીચે થોડું જવા દઈએ, લોગિન પર ક્લિક કરીએ. લોગિન પર સાઇટમાં છે ને. ઓકે, ઓકે. ઓલ રાઈટ, સી લોડ થવા દઈએ સાહેબ. હજુ હાલ લોગિન નથી કરવું સાહેબ, 1 મિનિટ વેઇટ કરીએ. પહેલા એકાઉન્ટ બનાવતા શીખવી લઈએ. સી થોડું નીચે લઈએ. મેં તમને બધાને સમજાવ્યું હતું, યસ એક્ઝેક્ટલી, કે 2 પ્રકારના ઓપ્શન છે. 2 પ્રકારના અહીંયા ભૂલ ક્યાં થાય છે એકાઉન્ટ બનાવતી વખતે? અહીંયા છે, જુઓ નીચે લખેલું છે કાઉન્સિલ સાહેબ, એના પર ક્લિક કરતા જઈએ એટલે ખાલી એના પર કર્સર લઈ જઈશું. યસ, કાઉન્સિલમાં રજિસ્ટર્ડ ફાર્માસિસ્ટ મતલબ જી નંબર અહીં એકાઉન્ટ બનાવવાની શરૂઆત 2 પ્રકારના લોકો માટે છે, 2 પ્રકારના ફાર્માસિસ્ટ મિત્રો માટે છે. એકાઉન્ટમાં જી નંબર ઓલરેડી છે, મતલબ તમારે બધાએ આ સાહેબે હમણાં કર્સર મૂક્યું હતું એ જગ્યાએ ક્લિક કરી અને એકાઉન્ટ બનાવવાનું છે. જ્યારે બાજુનો એકાઉન્ટ નો ઓપ્શન છે એ જે લોકો હમણાં અભ્યાસ પૂરો કર્યો છે અને એમનું રજીસ્ટ્રેશન કરવાનું બાકી છે, મતલબ રજીસ્ટ્રેશન ની અરજી કરે છે, રજીસ્ટ્રેશન નવું રજીસ્ટ્રેશન મેળવવા માટે જેને અરજી કરવાની છે એ લોકો માટે અહીંયા ભૂલ નહીં કરો તો બાકીનું બધું સરળ છે મિત્રો. અહીંયા ભૂલ કરી દેશો તો તમારે અમારો કોન્ટેક્ટ કરવો પડશે અને અમારે સોફ્ટવેર વાળા મિત્રોની મદદ લઈ અને તમારું એકાઉન્ટ સેટલ કરવું પડશે. અધરવાઇઝ જો અહીંથી તમે રાઈટ સિલેક્ટ કરી લીધું, ઉસ્માન ગની સર એકાઉન્ટ બનાવવાના છે અથવા મારે હું ઓલરેડી રજીસ્ટર્ડ છું અને મારે એકાઉન્ટ બનાવવું છે, એના પર ક્લિક કરીએ સાહેબ. ચલો આના પર ક્લિક કરશો, તમારું એકાઉન્ટ કઈ રીતે બનશે ભલે વિલ ટેક 5 મિનિટ્સ, પણ મને લાગે છે કે મારે એન્ટાયર પ્રોસેસ સમજાવી દેવી જોઈએ. ઓકે સર, એના પર ક્લિક કરીએ. હા યસ, આ વિડીયો છે, જે પ્રોસેસ હું સમજાવી રહ્યો છું. કેટલાક મિત્રોને હજુ પણ પ્રોબ્લેમ હોય તો એના માટે એક વિડીયો પણ મૂકેલો છે, એ આખો એન્ટાયર તમે સમજી શકો છો સાહેબ. એને ફાસ્ટ ફોરવર્ડ કરી દઈએ. યસ, ઓકે. હા, આખો વિડીયો પણ મૂક્યો છે મિત્રો તમારી સમજ માટે. ક્યાંય જૂના ફાર્માસિસ્ટ મિત્રોને નથી ખ્યાલ આવતો તેમની સમજ. વિડીયો પૂરો થાય છે એના પછી રિન્યુ રીએન્ટ્રી ઉપર ક્લિક કરવાનું છે ઉપર બ્લેકમાં. યસ એક્ઝેક્ટલી, આ પેજ ઉપર આવીને તમારી ડિટેલ ભરવાની છે મિત્રો. તમારું નામ છે, ડેટ ઓફ બર્થ છે, તમારો રજીસ્ટ્રેશન નંબર ધેટ ઇઝ યોર જી નંબર. ઈમેલ એડ્રેસ સૌથી ઇમ્પોર્ટન્ટ છે મિત્રો. ઈમેલ એડ્રેસ તમારે એ લખવાનું છે કે જે વર્કિંગ છે, ટાઈપ કરવામાં પણ ભૂલ ન થાય એટલા માટે આ ઈમેલ એડ્રેસ છે ને એ તમારું લોગિન આઈડી થઈ જવાનું છે. જ્યારે પણ તમારે વેબસાઇટમાં અમારા લોગિન કરવું પડશે તો લોગિનના 2 ક્રેડેન્શિયલ છે, આઈડી અને પાસવર્ડ. આઈડી એ તમારું અહીંયા જે ઈમેલ આઈડી લખવાના છો એ થવાનું છે મિત્રો. અને બીજું કે ઈમેલ આઈડી ટાઈપ કરવામાં ભૂલ એટલા માટે ન કરીએ કે જ્યારે તમે પાસવર્ડ ભૂલી જશો તો રીસેટ કરવા માટેની લિંક તમારા આ ઈમેલ આઈડી પર જવાની છે. તો દેટ ઇઝ મોર ઇમ્પોર્ટન્ટ. તો મેક શ્યોર કે તમારું જે આઈડી યુઝ થાય છે એ લખીએ. દેન યુ હેવ ટુ ક્રિએટ યોર પાસવર્ડ, મોબાઈલ નંબર અને પાસવર્ડ. પાસવર્ડ 8 આંકડાનો કરવાનો છે, એનીથિંગ યુ કેન ક્રિએટ. અને પછી ક્રિએટ એકાઉન્ટ કરવાનું છે સાહેબ. ક્રિએટ પર કરીશું એટલે એકાઉન્ટ તમારું બની જશે. મતલબ કે ઉસ્માન ગની સાહેબે આ ડિટેલ્સ બધી એડ કરી લીધી અને હવે એમનું એકાઉન્ટ બની ગયું. હવે એ લોગિન કરશે. તમારું આટલું ભર્યા પછી ક્રિએટ એકાઉન્ટ કર્યા પછી તમારે લોગિન કરવાનું. સી એઝ આઈ સેડ, લોગિનમાં તમારું ઈમેલ આઈડી એ તમારું લોગિન આઈડી થવાનું છે. તો એ યાદ રાખવાનું છે. સાહેબનું એકાઉન્ટ આપણે ખોલીએ છીએ, એ આઈડી અને સાહેબ પાસવર્ડ એડ કરશે એટલે એમનું એકાઉન્ટ બનશે. અને આ એકાઉન્ટ બન્યા પછી તમારી પ્રક્રિયાઓ શું છે? મેં કહ્યું હમણાં રીએન્ટ્રી કરવાની થશે. તમે રિન્યુઅલનો ટાઈમ મિસ કરી દીધો, ડ્યુ પીરિયડ ગયો, મતલબ ભલે 6 મહિના માટે ગયો, 12 મહિના કે 3 વર્ષ કે 4 વર્ષ રિન્યુ નથી કર્યું, કઈ વાંધો નથી. તમે યુ કેન રી એન્ટર યોર નેમ ઇન ધ રજિસ્ટર ઓફ સ્ટેટ ફાર્મસી કાઉન્સિલ. તો એ એપ્લિકેશનનું નામ છે રી એન્ટ્રી, અને રી એન્ટ્રી પણ ઓનલાઈન કરવાની છે. એવરીથિંગ હેઝ ટુ બી ડન ઓનલાઈન. તો આ પેજ સાહેબ ઓપન થાય એટલે હું તમને સમજાવું છું કે ક્યાંથી. વોટ ઇફ યુ આર નોટ એમ્પ્લોયડ, જસ્ટ રિઝાઇન્ડ. મિત્રો હા, રી એન્ટ્રીમાં એક ઓપ્શન આપણે માંગી રહ્યા છીએ કે તમારું જોબ પ્રૂફ. તો જોબ પ્રૂફ તમારે મૂકવાનું છે, જ્યાં કામ કરી રહ્યા છો એનો પુરાવો મૂકવાનો છે. કોઈકે પૂછ્યું કે જસ્ટ રિઝાઇન કર્યું હોય તો રેઝિગ્નેશન લેટર તમારે અપલોડ કરવાનું છે કે વી આર નોટ વર્કિંગ. અને કોઈ હાઉસ વાઇફ છે, બિલકુલ કામ નથી કરતા, તો એવા લોકોએ પણ એક પ્લેન પેપર ઉપર એવું રાઇટિંગમાં લખીને આપવાનું છે કે આઈ એમ નોટ વર્કિંગ રાઇટ નાઉ અને આઈ એમ જસ્ટ અ હાઉસ વાઇફ કે જે વોટ એવર, એ લખી અને તમારે એમાં જોબ પ્રૂફમાં અપલોડ કરવાનું થશે. તો મિત્રો ઇવન ઇફ ઇટ ઇઝ નોટ લોડિંગ, આના પછીની પ્રોસેસ બહુ સિમ્પલ છે. તમે આના પછી એકાઉન્ટ બનાવીને જશો તો એના ડેશબોર્ડ ઉપર એપ્લિકેશન્સ બતાવશે કે રિન્યુઅલ છે, રી એન્ટ્રી છે, ગુડ સ્ટેન્ડિંગ છે, ડિગ્રી એડિશન છે, તમારું એડ્રેસ ચેન્જ છે, આ બધું છે. તો ખાલી એના પર ક્લિક કરી અને તમારા ડોક્યુમેન્ટ જે માંગ્યા છે અપલોડ કરવાના છે. તો મિત્રો તમારું એકાઉન્ટ આ રીતે બની જાય. કદાચ અત્યારે અમારું સર્વર સાહેબ એના પર કામ પણ ચાલી રહ્યું હતું, તો સ્ટેટ કાઉન્સિલનું સર્વર ડાઉન હશે. વી આર અપગ્રેડિંગ શનિ રવિ, એવું કારણ હોઈ શકે. તો ચિંતાની જરૂર નથી, પરંતુ એક એકાઉન્ટ બનાવતા શીખવી દીધું છે, ધેટ ઇઝ ઓકે. તો શેરિંગ બંધ કરીએ સાહેબ, વાંધો નહીં. તો મિત્રો તમે બધા એકાઉન્ટ બનાવ્યા પછી લેટ મી નો અબાઉટ વિચ રજિસ્ટ્રેશન. તમે એક વખતે એકાઉન્ટ બનાવી લેશો પછીની બધી પ્રક્રિયા ઓનલાઈન ઈઝી છે. રિન્યુઅલ તો બહુ સરળ છે મિત્રો, અને રિન્યુઅલ થયા પછી આ જ વેબસાઇટમાં ડેશબોર્ડ ઉપર એક ડાઉનલોડનું બટન આવશે. એમાં તમારા બધી જ ફી ભર્યાની પહોંચ આજ પછીની એમાં રહેવાની છે. તમારી બધી જ રિન્યુઅલ અથવા રીએન્ટ્રીની સર્ટિફિકેટ્સ બધા એમાં રહેવાના છે. તો આ તમારું એકાઉન્ટ સ્ટેટ કાઉન્સિલની અંદર એ બહુ અગત્યનું છે, અને એના આઈડી પાસવર્ડ યાદ રાખવા બહુ જરૂરી છે. એક વાર એ થઈ જાય છે પછી ઘરે બેઠા જેમ રિફ્રેશર કર્યો છે એવી જ રીતે ઘરે બેઠા રિન્યુઅલ, ઘરે બેઠા રીએન્ટ્રી અથવા તો બાકીની બધી પ્રક્રિયા છે. તમારું એડ્રેસ ચેન્જ, એવરીથિંગ ઇઝ વેરી ઇઝી. તો મને લાગે છે કે બધાને આઈ હેવ એક્સપ્લેન્ડ ધ થિંગ્સ વિચ હેઝ ટુ બી ડન, and would like to conclude my speech once again thanking Dr. Usman Ghani sir and his entire team, principal sir and entire college and all the participants. Thank you very much sir. રજીસ્ટ્રેશન સર્ટિફિકેટ કયું એટેચ કરવાનું એવા પ્રશ્નો છે. તો આપણું રજીસ્ટ્રેશન સર્ટિફિકેટ સ્ટેટ ફાર્મસી કાઉન્સિલમાં તમે રજીસ્ટ્રેશન કર્યું છે અને જે રજીસ્ટ્રેશન સર્ટિફિકેટ અમે આપીએ છીએ એ એટેચ કરવાનું છે. ડિગ્રી સર્ટિફિકેટ એટેચ એ કરવાનું છે જે તમે કોલેજમાંથી પૂર્ણ કર્યા પછી લોકો પ્રોવિઝનલ મળે છે, પછી એનું ફાઇનલ ડિગ્રી સર્ટિફિકેટ મળે છે, પણ એ ક્યારેય અમારા રેકોર્ડમાં અપડેટ કરતા નથી. એટલે અમે વી આર આસ્કિંગ ફોર ધ ડિગ્રી સર્ટિફિકેટ. તો તમારે એ ડિગ્રી સર્ટિફિકેટ અમારા રીએન્ટ્રી વખતે એટેચ કરવાનું થશે. આ બધા ડોક્યુમેન્ટ રીએન્ટ્રીમાં માંગે છે. રિન્યુમાં ઓનલી રિફ્રેશર કોર્સ સર્ટિફિકેટ એન્ડ યોર આધાર કાર્ડ. હવે પછીના રિન્યુઅલ્સમાં અમે એ પણ અપગ્રેડ કરી રહ્યા છીએ કે આ જે રિફ્રેશર કોર્સ થઈ રહ્યા છે એ ઇન્ટિગ્રેટ કરીશું. તમે કર્યો છો, તમારી હાજરી અહીંયા છે, તો સર્ટિફિકેટ પણ અપલોડ કરવાનું નથી, સીધું જ અમારા રેકોર્ડમાં આવે છે. ઓનલી આધાર કાર્ડ. આ વખતે આધાર કાર્ડ અપલોડ કર્યું તો નેક્સ્ટ ટાઈમ આવશો ત્યારે તો આધાર કાર્ડ પણ નહીં, જસ્ટ પે ધ ફીસ, ક્લિક ઇટ, ગેટ રિન્યુડ. આટલું બહુ સરળ થવાનું છે. પરંતુ હવે અપગ્રેડેશનમાં છે, આટલા તો પહોંચ્યા છીએ. આવનારા 1 year પછી તો તમારો રિફ્રેશર કોર્સ તમે એટેન્ડ કર્યો છે, સર્ટિફિકેટ પણ એટેચ કરવાની જરૂર નથી. તો અહીંના સીધા જ કોલેજના ડેટા અમારા સર્વર પર ચડી જશે અને ત્યાંથી જ તમારું ક્લિક થઈ જશે કે મિસ્ટર XYZ રિફ્રેશર કોર્સ આ તારીખે અહીંયા કરેલો છે, એટલે તમારે એનું સર્ટિફિકેટ પણ મૂકવાની જરૂર નથી. થેન્ક યુ વેરી મચ સર.
થેન્ક યુ સો મચ સર. આઈ થિંક બધા પાર્ટિસિપન્ટ્સ ડાઉટ કદાચ ક્લિયર થઈ ગયા હશે તમારી ઇન્ફોર્મેશનથી. નાઉ આઈ વુડ લાઈક ટુ ઇન્વાઇટ કોઓર્ડિનેટર ઓફ ટુડેઝ રિફ્રેશર કોર્સ, Dr. Usman Ghani Chalotia sir, Associate Professor, IICP College of Pharmacy, to present a vote of thanks. Usman sir.
Yeah. Thank you, madam. My voice is audible.
Oh, yeah.
Yes sir, it's audible.
Yeah. Good noon to all distinguished guest speakers and dear participants. On behalf of organizing committee of IICP College of Pharmacy, Constitute College of the CVM University, I propose a vote of thanks on this historic occasion. I would like to express my appreciation for dedicated participation in the event who has come to end, and the event like this cannot be possible without your support. To all the person, I would like to take a minute to thank all of them. The online refresher course was an overwhelming success, attracting more than 200 delegates, and speakers provide a great intellectual and social interaction for the participants. We are heartily thankful to management, Charutya Vidya Mandal, the Charutya Vidya Mandal University, and Gujarat State Pharmacy Council for giving wholehearted support for this event. We are thankful to our Chairman, Engineer Bhikbhai Patel sir, Chairman of CVM and President of the CVM University, our Chief Guest, Dr. Srikant Joshi sir, Executive Committee Member of the Gujarat State Pharmacy Council, Ahmedabad, and Associate Professor, Department of Pharmacology, Maliba College of Pharmacy, Bardoli, Surat, for delivering informative knowledge about the role of pharmacy and recent activity of GSPC. I'm also thankful to our another chief guest, Provost Dr. Himanshu Soni sir, the CVM University, for his encouraging words. We are thankful to all our resource persons for accepting our invitation, sharing their valuable experience and knowledge in the event. I would like to extend my gratitude to our principal, Madam Dr. Harshavi Patel madam, for taking efforts for making this refresher course possible. I'm very thankful to all teaching, non-teaching staff for their direct and indirect support for making this event successful. I'm especially thankful Ms. Heta Kachya, Ms. Jinal Tandil, Ms. Mittal Verma, and Mr. Kunal Solanki and Mr. Dhaval Patel for putting their great efforts for handling MOC and IT related activities. All the audience, thank you for paying your valuable attention, and kindly suggest your opinion and on online feedback and making the event successful with your contribution. thanking you with your blessing with your presence. Now I would like to officially end this event. I'm very thankful to all of you. One more thing, we are putting a feedback as well as the link of objective test, which is working for half an hour. So kindly both the things, both the individual link you have to fill feedback as well as the online objective test within a due time and submit it by everyone. So I will keep it this link in this chat box as well as the WhatsApp group. So thank you very much, everyone. સી તમામ ફાર્માસિસ્ટ મિત્રોને જણાવવાનું કે હાફ એન આવરમાં ફીડબેક ફોર્મ અને ઓબ્જેક્ટિવ ટેસ્ટની ઇન્ડિવિજ્યુઅલ લિંક જે આપેલી છે એ ફિલઅપ કરી અને સબમિટ કરે. જો ઓબ્જેક્ટિવ ટેસ્ટની લિંક યા ટેસ્ટ નહીં આપ્યો હોય યા ફીડબેક ફોર્મ નહીં ભર્યું હોય તો તમારું સર્ટિફિકેટ ઇશ્યુ નહીં થાય. લિંક માત્ર હાફ એન આવર માટે વર્કિંગ છે બંને. તો વિધિન 30 મિનિટ્સમાં ઓબ્જેક્ટિવ ટેસ્ટ અને ફીડબેક ફોર્મ તમે ભરી દો. નાવ આપ સૌ આ મીટિંગમાંથી લેફ્ટ થઈ શકો છો. We are ending this Zoom meeting within 2 minutes. સારું ચલો કાલે જોઈએ હવે. બસ તૈયારી કરીને આવજો તો નાસ્તો આપતો લઈને આવજો.